So, a very good morning from Amsterdam. I'm Emer Cook. I'm the Executive Director of the European Medicines Agency, and it's a real pleasure to welcome you here on behalf of the EMA and the European Regulatory Network. This conference is designed to promote the development of RNA-based medicines, and we have had an unprecedented interest with more than 1,000 registered participants and many more following online that we couldn't accommodate as registrants. So participants in today's meeting include academia, regulators, industry, both big pharma and small and medium-sized enterprises, the European Commission, DG Research, patient and healthcare professional representatives, and of course, many other speakers, including some joining from overseas at a very difficult early time for them. Now, as you all know, the COVID pandemic has been a high burden to most of us, both professionally and personally, over the last 3 years. But today we want to explore some of the possibilities arising out of these challenging times. mRNA technology has been around for a long time, and we knew there was great potential, but the development and evaluation and application of mRNA vaccines during the pandemic really served to translate that promise into reality. So the progress made over the past three years in mRNA and related technology is really unprecedented. So we need to ensure that the knowledge gained from these advances can now be put to the best use for drug development, which will benefit patients for the future. As far back as 2012, the European Medicines Agency and the EU regulatory network first looked at some advances in RNA technology, in the technology that has now been used in COVID vaccines. And at that time, that was by an academic group, and it was for the treatment of cancer. So we need to understand why those developments did not translate into products at that time, and is now the right time. How do we move from failure to success with many other products? mRNA technology provides us with a great example and a success story. But the focus of this conference actually goes beyond mRNA, and my colleague Stefan Thurstup, who's our Chief Medical Officer here at EMA, will elaborate further on this. So the conference will cover a large range of topics and areas of drug development, and we probably won't be able to go into in-depth discussions in all areas. So what we'll do is we'll identify some of the areas and we'll follow them up afterwards. I want to say a special thanks to all the speakers, to the chairs, to the organizing committee for their engagement and commitment. I think this is a really important conference, and I wish you a very inspiring day, and I'm really looking forward to tangible outcomes that lead to benefits for European patients. Thank you very much.
Many thanks, Emer, for this warm welcome, and warm welcome to all of you who join us either on WebEx or for the live streaming. I'm Stefan Thurstup. I'm the Chief Medical Officer here. I will be chairing this session together with Sol Ruiz. Sol is working for the Spanish agency and also chair of the CHMP Biologicals Working Party. And Sol is with us online. Good morning, Sol. So thanks to everybody who joins this conference. I hope we will be able to have a very interesting day. As already alluded to by Emer, this conference will not be able to go into each and every technical detail of each and every existing or future RNA technology. The idea is to stimulate development of these RNA therapies and to engage with the academic and pharma companies, with EMA and the regulatory network. The ultimate scope of the conference is to identify research needs, regulatory challenges, and opportunities in this field. I will stress that we deliberately have taken vaccines for infectious diseases out of the scope. mRNA will be dealt with, but outside the infectious disease area. We will also leave out discussion of RNA used for gene editing technologies. Another important issue is that we will be strict on timing. We have a very dense program, and we hope that we'll be able to leave room for Q&A at the end of each session. At the end of the day, at session 5, we will have a more forward-looking panel discussion focusing on areas within the scope of this conference. Questions you may have can be submitted via Slido, and all registered participants received yesterday an email with the code to Slido so they can access and put in their questions in writing. More practicalities is that this session or this day will be recorded. Recording will be made available after the conference on our website, and the same will the slides and ultimately a report with the outcome of this session. Before I hand over to my colleague Frank Immen for the first session, I think it would be important for us and for you who participate to get an idea of who we are, who is actually online now. Close to 500 participants has already logged in. So please, here is a poll where you can answer some questions that will give you an idea of who you are. I hope you are all able to see the now things are happening here. Yeah. I know that not all 500 have voted yes yet, but it at least gives you all a flavor of who you are together with today in this virtual room. A majority of pharma industry, almost a little over half of you, 2/3 maybe, a lot of regulators, and my impression is that we are talking about not only regulators from Europe, but from across the world. So this is really an international thing. Also nice to see academic and patient organizations and healthcare professionals joining here. We will let the poll continue a little bit further, and we can have this on your expertise. Also nice to see a broad representation, especially I hope more clinicians will join later on when we get to the clinical session, but let's see. At least thanks for this. And do we have a last one? No, that was the last question, sorry. So by this, thank you for joining once again. Hope you will have a very fruitful outcome of this day, and I'll pass on to Falk to introduce the first session. Thank you.
Thank you very much, Ima and Stefan, for this nice introduction, and also welcome all the participants joining us. I have the pleasure of moderating the first session, State-of-the-Art RNA Technologies, Setting the Scene. We have 3 great speakers with background from academia, regulatory, and industry. We'll run the 3 presentations back to back, taking questions and answers after the third talk. For the stakeholders joining us via WebEx, we will now open as well the possibility for you to submit the questions. So please do this throughout the presentations, that we have a wealth of questions when we start the question and answer session. So it's my pleasure to introduce now Sol Ruiz. She heads the biotech office at the Spanish Medicines Agency, is also responsible for ATMP, so cell, gene, and tissue engineered products. She's currently chairing the Biologics Working Party at the European Medicines Agency and is also a member of our Committee for Advanced Therapy and a member of our most relevant committee, the Committee for Human Medicinal Products. Sol, thank you very much for agreeing to speak.
Thank you. Thank you so much to you, Falk. I hope you can hear me well. Thanks for inviting me to be here, and thanks for your introduction. So let's see if we can get the presentation. And I know we will be strict with time, so please, Falk, let me know if I, get close to the last minute. Thank you. Thank you so much. Okay, so we will start with providing a very brief regulatory perspective on this class of medicinal products. If we can go to the next slide, please. Next. Sorry for the animation. So what I would like to go through in the next few slides is like, what do we have now regarding RNA-based medicinal products? What products have been approved? What are we expecting in the near future? And some of the regulatory challenges that we may face. Of course, I mean, how can we provide support during the development, and hopefully providing also basis for international harmonization. Next slide, please. So this is the topic of the conference today. So we can find, mRNA molecules or non-coding RNAs. Next slide, please, or next 3, if you don't mind. So we have mRNA in therapeutics, we have mRNA vaccines. As Stefan mentioned, we will not deal today with vaccines against infectious diseases, but we have others used as therapeutics. We also have molecules, as you can see, antisense oligonucleotides, RNA interference, and I will not go into this in detail, as we will have, we will go see them in following presentations. So next, please. So we have a very well-established and clear regulatory framework for medicines in the European Union. And of course, we are all very familiar with molecules that are chemically synthesized, used in many different therapeutic areas. Next, please. And but also we have many biological medicinal products that are obtained from a biological source and for which the production process and control may have an impact on the efficacy or safety of the product. So among these, next slide, please. We have immunological medicinal products such as vaccines with a well-established regulatory framework, large experience. We have other products such as allergens that we have we're less experienced and maybe more dealt at the national level. Next, please. We have also medicinal products obtained from next, please, from human blood and plasma, such as coagulation factors, immunoglobulins, or albumins, but we have others. And if we go to the next, please, and maybe another one, please, next. We have many medicinal products obtained by genetic engineering, biotech products obtained in different expression systems, such as mammalian or prokaryotic cells, but also obtained in transgenic animals with different types of molecules, such as hormones, enzymes, monoclonal antibodies, or fusion proteins. But also, next, please, we have advanced therapy medicinal products, including gene therapy, cell therapy, and tissue engineering. Next, please. So we have biotech products, such as recombinant proteins that are obtained in genetically modified cells or organisms based on the information that is in the DNA and where the specific gene of interest has been introduced. We also have some experience with using the DNA itself, such as from gene therapy medicinal products, very small experience or limited with plasmid DNA vaccines. There's one COVID-19 vaccine authorized in India, and we have several vaccines both authorized in the EU or in the US for veterinary use. Next, please. And this is the new class of molecules, as Emma also mentioned at the beginning, at the introduction, that we have gained quite some experience in a very short time frame. And we will see, obviously, more products coming very soon. Next, please. So obviously, the experience, even though mRNA vaccines are excluded from the scope of this meeting, but obviously the success that we have had with COVID-19 vaccine has made or has marked the beginning of a new era in modern medicine. So obviously, what we have learned in all areas, quality, non-clinical and clinical, with COVID-19 vaccines, we can use it as a basis to move forward to other RNA-based medicinal products used in different areas. Next one, please. And similarly to what we have seen with other medicinal products, as you can see in this review from 2001, if we go to the blue box, and particularly the line that says all monoclonal antibodies, we can see that only 10 monoclonal antibody therapeutics were authorized between 180 and 2000. Next one, please. But currently, at the end of January, we have already 126 monoclonal antibodies approved. In the EU, 36 are biosimilar molecules, and in many different therapeutic applications, as you can see, hypercholesterolemia, arthritis, but also multiple sclerosis, migraine, hemophilia, etc. Next, please. Similarly, something similar happened also in the field of gene therapy, where you can see that the first clinical trials started in the late 80s, early 90s, and then at the end of 2018, we have had a steady, progression in the number of clinical trials. Next, please. To get to today, where we have 25 advanced therapy medicinal products authorized in the EU. So I think we will see something similar with RNA. The experience with mRNA COVID-19 vaccines has really set a very good basis to get new products on the market. Next, please. So when we are in here, please, if you can do, I think it's 2, 3, maybe next. To get the animation, we have products, as you can see, in green, products that have been approved, in yellow, under development, in orange, that have been withdrawn. We have mRNA-based medicinal products such as mRNA molecules used for the treatment of different oncology indications, also used for to obtain CAR T cells, either in vivo or ex vivo. We have antisense oligonucleotides approved for different indications, as you can see. And here, we have a sort of a first challenge. I would not say it's very big, but one of the issues may be that depending on how the product is obtained, can be classified as a chemical, as a biological molecule, or as an advanced therapy medicinal product. Next slide, please. So again, other examples of RNA interference, small interfering RNAs used in different indications, as you can see, that have been approved, but also others under development. And as mentioned at the beginning, we will leave out of the scope RNA used for genome editing during this meeting. So again, very interesting products in many different applications that we will see in the near future. Next, please. So these are the numbers that we have so far at the EMA. So as you can see, 95 initial scientific advice applications, 50 initial protocol assistance, some also with follow-up assistance, 110 for orphan designation, or some others also to maintain to keep this orphan designation, 24 pediatric investigation plans, and already 13 applications for marketing authorization, where 10 have already considered valid. So again, we will see very soon these products that will be under evaluation. Next, please. So very brief conclusions from what we have seen approved and what is coming. So I think mRNA medicines are well included in the current regulatory framework. We may need some adaptations regarding the definitions, and this might be on the way. We can certainly build from experience, and specific guidance will be under development soon. We will see in the next presentations. And obviously, regulatory support is available at any stage of development. If we go to the next slide, please. You can find this booklet on the EMA website on how you can move from initial research till the medicine is on the market. Next. And as we always say, next, please. In this, long and winding road, obviously, national agencies and also EMA can provide a very useful advice that can help you to do this route. Next, please. So support can be obtained from product classification. If you're an SME or you have an orphan indication, there is incentives, or if you're eligible for Prime. Next, please. And then you can always go, next, please, through the EMA Innovation Task Force, where they will be very helpful. But next, please, is also very useful to go through the European Public Assessment reports for products that you consider are similar to the one that you're developing. There's very useful information on quality, non-clinical and clinical aspects. Next, please. And obviously, yeah, there's very important that regulators are talking to each other, and there are projects to increase this dialogue with other agencies. Next, please. And we'll go to the last. So we know you're all the Edmund Hillary's trying to reach the top, and we would like to see ourselves as the Sherpas in Norway trying to help to make this road as efficient and quick as possible. Thank you. Thank you so much for your attention.
Thank you very much, Sol, for putting RNA-based medicines into context of other success stories we saw as regulators, and also highlighting recent successes, raising interest and development of these medicines for the benefit of patients. So it's my pleasure now to announce our speaker, Tal Sachs, who is adjunct associate professor of medicine at the University of Pennsylvania. Tal was recently chief medical officer at Moderna, where he led the development of the COVID vaccines, beside other key programs. He's also partner of OBIMed. And special thanks, Tal, for supporting this conference at so early hours. Thank you.
Thank you. So good morning, everyone. My name is Tal Sachs. It's a real pleasure to be here to provide a perspective from industry into this important conference. I apologize for my hairdo. It's a function of the hour. Let me begin by thanking EMA for hosting this important meeting and the organizers for inviting me. I've been closely involved with RNA medicines by virtue of my prior role as the Chief Medical Officer of Moderna, and also my current role as a partner at OBIMed, a venture capital firm, where I'm happy to say we're investing in several companies in this field. On the next slide, you can see my disclosures. And if we go to the next slide, I think while we're all starting from well understood definitions and opinions as laid down in regulatory guidance and precedent, if we are to improve, then it's critical we ask ourselves, what are the exceptions that point to these challenges? And that's what I'm going to try to do in some of my opening remarks. At the highest level of definitions, I think the overarching question in my mind and that of my colleagues is whether, just like biologics emerged a few decades ago as a novel class of medicine, perhaps RNA medicines are ready for a prime time designation as their own class alongside novel chemical entities and biologics. So on the next slide, from an industry perspective, what is it that we care about? Well, the first is to have frameworks so that we know what to expect and we can tailor our activities to meet those expectations. And from a clinical perspective, while we all understand the fundamental paradigm of demonstrating benefit risk, I think mRNA medicines do pose unique challenges because the pharmacology here is a function of both the sequence or the information that the drug encodes, as well as its chemical or biological backbone. And this sequence-based characteristics, whereby drug candidates share a platform backbone but differ in the information they encode, actually enables rapid progress. Between November 2015 and November 2020, Moderna went from being a preclinical company to having over a dozen of drug candidates in the clinic and its first product deployed to millions of people. Now, of course, the pandemic played a role, but it was the underlying technology that enabled this rapid response. Our ability to understand what safety attributes are likely platform mediated versus those that would be sequence dependent has been crucial to the success of the COVID vaccine. The second thing we care about is whether the same preclinical and clinical data can be globally useful. However, as in any new emerging modalities, different regulators sometimes have different opinions as relates to some of these critical attributes. Even something that sounds straightforward, like what is gene therapy, is getting very different treatment. As this class matures, I hope we find ways to harmonize our approaches. And finally, we care deeply about what our stakeholders make of our effort. And I think this is especially challenging when we talk about RNA medicine. All of us have deep technical expertise and find it easy to communicate amongst ourselves. But without the broad public understanding and accepting of what it is that we do, largely on their behalf, whether as civil servants or in the commercial space, our efforts may be in vain. And I'll come back to this point at the end. Next slide, please. Dr. Ansmarus will review this class, and so I don't want to get into the details here, but what I do want to do is point to the fact that actually RNA medicine encompasses a very diverse set of pharmacological interventions. And as I said, the unique challenge here is because the action is exquisitely dependent on both the sequence and information encoded in the molecule, as well as its backbone and where it goes. These medicines need to get inside the cell and sometimes even inside the nucleus. So their benefit risk is ultimately also dependent on the delivery vehicle or modality. And I'll start with the obvious that I'm sure will be discussed later today. These emerging technologies do not necessarily neatly fit into our categories or into any one-size-fits-all box, frankly. We may think of short oligos as being more like small chemical entities and mRNA as being a biologic, but it's entirely conceivable with current technology that one could synthesize an mRNA molecule without the use of a plasmid, so suddenly it would be a chemical entity, and under current regulations, no longer gene therapy. The second question is whether gene therapy should be defined by the fact that the drug is made up of nucleic acid. Should it be defined as whether or not those nucleic acids were put together based on a biologically derived template? Or maybe it should be defined based on the intended or observed pharmacological action, and further or not, whether this action is anticipated or has a scientific basis to have replicative potential. And this last point would make, in terms of definition, relates to this notion of a specific platform. While mRNA does not reach the nucleus or directly affect the DNA, of course, mRNA could encode for a nucleus that would get into the DNA. And this distinction is critical when we think of what's required in terms of clinical safety assessment. So I think the concept of a platform is warranted because it could enable us to make better extrapolations of anticipated benefit risk, both from preclinical to clinical and between one product to another, giving the scientific similarity. This has obvious implications that I'm sure others will cover, especially as it relates to non-clinical toxicology. And, especially in this day and age, with the sensitivity around the use of non-human primates to guide clinical experimentation. But it may also have important implications for the level of evidence that we should expect prior to authorization, and perhaps in an era where the emergence of real-world evidence as a basis upon which one can actually draw inferences in this day and age of digital clinical data collection, as we've seen and leveraged very effectively in the development of the COVID vaccines, we can think of more creative ways in which, when we have a platform and we have better understood anticipation of benefit risk, we can better leverage data collection in real-world evidence post-approval, as opposed to insisting that everything be checkbox pre-approval. So on the next slide, if you'll allow me to double-click on this notion of a platform, I think we already have examples of this. When we developed the first lipid nanoparticle encapsulated mRNA that led to a potential therapeutic levels of a translated monoclonal antibody, even in phase 1, because we were in a lipid nanoparticle, both we as a sponsor, I as a chief medical officer, and obviously the regulators were informed by the prior experience of a very different class of RNA medicines, the siRNAs, that had already been successfully developed within a lipid nanoparticle. Now, that being said, I fully expect that from a safety as well as efficacy profile, the same LNP may complex differently with an mRNA as it would with a different type of mRNA. So clearly there are similarities, yet the level of evidence will need to be appropriately obtained. And I think perhaps the best example of this, both the benefit and challenge of this platform, is that of a personalized cancer vaccine, a therapeutic cancer vaccine, which has recently been demonstrated to have very interesting potential in a randomized phase 2 setting. And on the next slide, as we consider the, I'm not sure the slide is advanced. Yes, perfect, thank you. So on the slide of the, if you can see what we're doing here, we're basically encoding a patient-specific different vaccine for every patient based on their tumor DNA. And it's an automated algorithm that decides for each patient what kind of mRNA they get. And that algorithm actually encompasses software in it. And this is software that is publicly available and gets updated. So here we have a medicine where it is essentially already a platform in and of itself, because every patient is going to get a different vaccine. And it's critical that as we think about this medicine, you know, when we designed it initially, the publicly available piece of software, it's called NetMHC, was in version 4 or 5. By the time the company gets to phase 3, it'll be version 6. Perhaps by the time it's actually deployed, there will be a version 7. And this is critical because as these versions improve based on more data, and that more data makes the prediction algorithms more accurate. And so how do we regulate this drug when it actually gets to the point of having demonstrated in a randomized phase 3 a benefit? Clearly, we've got a process, clearly we have an algorithm, but we want to make sure that indeed we're able to continue to improve that. And it's inconceivable that every time the software bit improves, we're going to have to run a new phase 3 trial. And so that for me is the edge case, if you will. And I think that this is an example where I would expect that, you know, real world evidence, once the vaccine is deployed in this day and age, should be able to play a critical role as we think about this. So let me close by coming back to the language we use for definitions. And I'm really happy to see that we have patient representatives and advocates in the audience. You know, the obvious challenge I personally faced when leading the development of the mRNA vaccine was that this notion of whether or not it's gene therapy. As mentioned by Dr. Ruiz, by EMA regulation it is, you know, but by the English language, and I put up here the definition from the Webster language, a gene is a functional unit of inheritance, which I don't think mRNA is. And this leads to confusion in the public. You know, in 2017, I gave a TEDx talk where I made the wrong analogy in describing mRNA. And during COVID, this became a source for both innocent and intentional misunderstanding of whether COVID vaccines alter the DNA, undoubtedly contributing to non-scientific vaccine hesitancy by perfectly rational people. And on the next slide, you know, if Reuters has to issue a press release to say that what people said you said has in fact been fact-checked and isn't so, well, you know, there's a problem. And I think one of the most important learnings for me from the success of the COVID vaccine is how critical it is that we in the technical domain really align our language and communication to enable the broadest of understanding. And let me close with that. Thank you for your attention. And I look forward to the sessions and any questions.
Thank you very much, Tal, also for raising relevant questions and challenges associated with RNA medicines. Also for flagging factors which are maybe not directly related to the research and development of these vaccines, but utmost important for successful implementation in the healthcare system, like your last 2 slides demonstrated, to really bring the medicines into the patients. It is my pleasure now to announce our third speaker. Mika Ansmarus is a professor of translational genetics at the Leiden University Medical Center. She co-founded the Dutch Center of RNA Therapeutics, aiming to develop exon skipping therapies for patients with unique mutations. She has received several awards for her contribution in the field of Duchenne muscular dystrophy. Thank you, Mika.
Okay, thank you for the introduction, Falk. What I will do is I will give a brief introduction to oligonucleotide therapies, focusing on single-stranded oligonucleotides. And I hope that those who are not, say, fully embedded in the oligo field will have some frame of reference after this talk. So next slide is my disclosure. If you want to read it at your leisure, take a screenshot now. Next slide. So what are antisense oligonucleotide drugs? They are small pieces of modified DNA or RNA. They can be single-stranded and double-stranded. I'll focus on the single-stranded ones here in this talk, but the siRNA that was mentioned already by both previous speakers, those are double-stranded oligonucleotide drugs. And as was already mentioned, they are synthesized from chemically modified nucleotides. So they're not biologicals, they are chemically synthesized compounds. And what makes them unique is that they target the RNA in a sequence-specific manner, so via Watson-Crick base pairing. And that means that they can intervene at the genetic level, and we can decide where they're going to target. And the aim that you can have with oligonucleotide drugs is twofold. You can get a knockdown of a toxic protein, or you can restore a missing protein. Or in the case of mRNA vaccines, you can express a protein that you then want an immune response against. So on the next slide. So as was mentioned already as well, these compounds are synthetically produced, but we need to give them specific criteria to make them drugs, because if you have regular RNA or DNA, that is very unstable. So if you inject it into the body, it will be gone in 10 seconds. So you need to improve the stability. So you need to make them resistant to nucleases, enzymes that break down RNA and DNA. And you can do that in different manners. You can modify the backbone of the oligonucleotide, and you can modify the sugar. And you can modify them in many ways. We're not going to go into the chemical structures, but what's important to know is that even if you modify them in such a way that they don't really look like DNA or RNA anymore, they still bind via Watson-Crick base pairing. And with the mRNA vaccines, you have very little opportunity to modify because it needs to be translated. But if you target RNA, like oligonucleotides do, then you have much more flexibility. The first modification is the phosphorothioate. That's a backbone modification. And what that does, it doesn't only improve stability, it also gives oligonucleotide bioavailability. You have to bear in mind that these oligonucleotides are very small. So normally they will be filtered out by the kidney because they are that small. Now, to prevent making really expensive urine, the phosphorothioate backbone will allow low affinity serum protein binding, and that prevents then renal clearance. Delivery can be systemic, and then the main target organs are the liver and the kidney, but most other organs do take up some oligonucleotides. If you want to target specifically to the liver, there is a conjugate that you can add, the GalNAc conjugate, and I'm sure it will come up later. However, if you deliver these oligonucleotides systemically, they will not go to the central nervous system. So they will not go to the brain, they will not go to the eye. So there you can do local treatment, intrathecal or intraocular. And I mentioned already, there's many chemical modifications available. The ones to use depends on your modality. So on the next slide. So I mentioned already, you can either decrease proteins or restore proteins. And why would you want to decrease a protein? Well, if there's a disease that is caused by a mutation that causes production of a toxic protein, for instance, Huntington is an example, then reducing the amount of toxic protein is expected to have a therapeutic effect. You can also target a key protein in a pathological pathway, and you can do this with RNase H and siRNA. And I mentioned already, I'll focus on RNase H here, but siRNA achieves the same thing, just with a double-stranded oligonucleotide and a different pathway. So on the next slide, I'll explain RNase H. So RNase H is an enzyme that we have in all our cells, which cleaves RNA, RNA, or RNA DNA hybrids. So if it recognizes a transcript where an antisense oligonucleotide is bound, it will cleave it, and then the transcript is cleaved, so the protein that it normally would produce cannot be produced. The challenge with RNase H oligonucleotides is that the enzyme binds on the sugar modification. So if you modify the sugar, this will not work. However, the phosphorothioate modification that I mentioned that you need for biostability or bioavailability, that is a double-edged sword, because on the one hand, it prevents renal clearance, but on the other hand, it is the main driver of toxicity. If you also modify the sugar, it reduces that toxicity. Also, it improves the affinity for your to your target. So what you want a gap where your RNase H enzyme can bind and cleave, but at the flanks you want to modify as much as you can to reduce the toxicity of the phosphorothioate and to improve the binding of the oligo to its target. On the next slide, there's currently 3 approved RNase-H oligonucleotides. Mipomersen for familial hypercholesterolemia, only approved in the FDA and has been taken off the market since. Inotersen for transthyretin-mediated amyloidosis. So that's a protein, or that's a disease caused by toxic protein. There's also an siRNA approved for the same disease, and then falonosorsen, and it's not for high cholesterol, but for a familial disease causing high triglyceride levels. What you have to bear in mind that if you target a key protein, you will reduce the amount of that protein. And it's, I mean, you have little control on how much decrease you have. If you target a toxic protein, usually it's not possible to only reduce the amount of toxic protein. So you reduce the amount of protein in general, both the toxic protein and the normal protein. So on the next slide, we move to protein restoration. So that works in a different way. That works by modulating the splicing. And I'll explain how that works on the next slide. And what's important to know here is these oligonucleotides do not activate RNase-H. They are fully modified, so both the ribose and the backbone is fully modified. And on the next slide, some basic biology on how splicing works. So the genes code for protein and the genetic code is dispersed over the gene. And these pieces of genetic code are called exons. In between are regions that do not contain protein code called introns. When a protein is made, first you get a complete copies, exons and introns, and then during splicing, all the exons are linked together. Now, what splice modulation does, it either wants to skip an exon that is included aberrantly, or when an exon is, due to mutation, no longer recognized, it wants to improve the recognizability of that exon. So you can do it either to include an exon that is missing or to exclude an exon that you don't want there. And on the next slide is outlined which drugs have been approved for this. So nusinersen is an example of an oligonucleotide that causes inclusion of an exon. It's approved for spinal muscular atrophy. And in the US, there's 4 exon skipping oligonucleotides approved for Duchenne muscular dystrophy, but only in the US, not in Europe. So on the next slide, something that is closer to my heart and the Dutch Center of RNA Therapeutics that was already introduced. So these splice modulation oligos give the opportunity to skip a specific type of mutation called the cryptic splicing mutation. And a cryptic splicing mutation is a mutation within an intron that suddenly makes part of an intron look like an exon. So it's included in the messenger RNA, but because it's not coding information, this messenger RNA does not encode a functional protein. If you could prevent inclusion of that exon, you will restore the normal protein. Now, if you have brain or eye diseases where you can do local delivery, this now offers an opportunity to have, say, individualized oligonucleotides. These mutations are generally very rare, so not that appealing to industry. So is there room for academics to develop these on an individualized basis? In the next slide is an example of exactly that. So Tim Yu in Boston's Children's Hospital in the US found a patient with a cryptic splicing mutation causing Betten's disease, and he designed oligonucleotides, tested them, had discussions with FDA, did toxicity studies, and within a year after finding the mutation, he injected intrathecally this compound into the spinal cord, or into the spinal cord. So the drug was called Milasen because the patient was called Mila. And based on this, so this inspired lots of people worldwide to do this, to develop these oligos for single patients. Of course, this needs to be streamlined, and there's lots of discussions that we can have about this. But what's good to know is that there is a lot of collaboration. So the mother of this patient, Mila, and the clinician doing it, Tim Yu, have set up the N of 1 Collaborative, which is a global network to coordinate the work. In the Netherlands, we have the Dutch Center for RNA Therapeutics, but also within Europe, we have the One Mutation, One Medicine. And well, the Innovation Task Force was mentioned. We went there as a DCRT and got very useful advice. We also went for scientific advice as 1M1M. Again, got very useful advice there as well. So the comment, come to the regulators early, it doesn't only apply to industry, it also applies to academic, and I can highly recommend it. On the next slide, we're coming back to oligonucleotides in general and the safety considerations. And these are not the chemically modifications that I'm relating to, but the safety considerations based on the targeted effect. So if you do knockdown, you target a specific transcript. And I mentioned already, often this is not allele specific. So you can get a saturated pharmacology, too much reduction of your protein, or reduction of your protein in the tissue where you want it, but also in the tissue where you don't want it. So you have to see if you don't want to knock something down in the brain and you target the liver, that's not so much of a problem because the oligonucleotides won't really go to brain unless you inject them locally. But these are things to consider and to think about. This is not an issue if you restore a protein, because if you restore a protein, the gene is under its natural promoter. And then off-target, and by off-target I mean minor mismatches where the oligonucleotide can bind even though you don't want it to bind there. How well oligonucleotides bind varies with specific chemical modification, but this is something that you can also check for in silico to make sure that your oligo is unique. So on my last slide, some summaries. So the drugs are coming of age, and was mentioned already, there's a lot more in the pipeline, so more will be coming. They can be used to knock down proteins or to restore proteins, and that gives them a very broad clinical applicability. And I think, well, the final thing that I didn't discuss here, but that is a point, delivery is challenging for some tissues. So there's a reason why there's certain type of diseases that are currently focusing, on, so such as brain and liver diseases. And that is my last slide. So I think the next one is the questions, and I give the floor back to Falk.
Thank you very much, Mika, for condensed insight into antisense oligonucleotides. Okay, we now start the question and answer part of the session. And colleagues, please be reminded who follows us via WebEx to submit your questions. We received a couple of questions on the classification, and we will take this as well. But of course, we want to have a wealth of questions as broad as possible. I'd like to start with maybe a more broader general question. What area, in your opinion, has the greatest potential materializing on recent advances in RNA technologies? And maybe I'd like to talk to start with Tal.
Well, you know, I think the greatest potential that I see is one that we can actually deliver mRNAs to more tissues where we currently are struggling. I think that is going to be one of the biggest enablers. So, you know, if we can get it really consistently into the brain, if we can get mRNA into the brain, you see experiments already with inhaled mRNA to the lung. So I think on the mRNA side, that's one aspect. The other aspect, of course, is using novel classes of RNA and even guide RNA that can get into the nucleus. And there is a potential convergence ultimately between elements of RNA technology and elements of gene therapy. And while I recognize gene therapy is out of scope for this meeting, I'm not so sure that in the future we'll be able to make such a clean distinction, and we'll need to pay attention to that. I see people taking very creative approaches to leveraging the utility of mRNA to actually encode components of gene therapy.
Thank you, Tal. This brings us indeed back to the classification. And Mika?
Yeah, so it was interesting because I was making a note while Tal was speaking that where we can deliver, that determines what it can be used for. And I think, well, currently there's a lot of potential for brain diseases, for liver diseases, because we can deliver there. But if we solve delivery to tissue X, Y, or Z, then suddenly that opens up a lot of opportunity for diseases of those tissues. So I think that is why delivery is important, and why improving delivery to different tissues is important. And I think specifically the oligonucleotides, so the gene targeting approaches, of course, for rare diseases, monogenic rare diseases, there's a lot of opportunities there, and currently still driven by where we can deliver, but the more tissues we can deliver to, the wider this applicability will become as well.
Thank you, Mika. Sol, as a regulator, you already showed us some of them. Please.
Yeah, no, I mean, fully agree with what Talg and Mika have mentioned. I think, we have seen what products are coming and in which areas. It seems that also, the use of RNA molecules in oncology and the possibilities of a personalized treatment, as Talg mentioned, I think it's really, we're seeing some, interesting results. And obviously also, as mentioned by Mika, I mean, in genome editing using RNA seems that is also very promising. So regarding some of the regulatory challenges, I think we take the message. I mean, we have identified this issue regarding classification, and even though from a requirement point of view, let's say what we will require from quality, excuse me, non-clinical or clinical perspective, I don't think it will be a big difference, but we have to be careful that, that the path is kind of efficient, that because of the classification, the product doesn't have any additional hurdles. But we will continue working on that. Hopefully, there will be a revision of the legislation and we can maybe shape some definitions. So already working on that. Thanks.
Thank you, Sol, for preempting this question already. We saw major advantages through the COVID-19 vaccines. And Mika, in your view, what is the major scientific or technological advantage or steps enabling really the development or the further development and treatment of patients with RNA-based medicines?
Yeah, so I think so. For society, it sometimes may seem like these vaccines, they came out of nothing, like there was nothing and suddenly there was mRNA vaccines. But I've been in the oligonucleotide field for over 20 years and I know it didn't came from nothing. There was a lot of preliminary work leading up to that and facilitating that when we needed it, the development could be quick. I think looking from an oligonucleotide therapy perspective, so not just the mRNA, but also targeting the mRNA, I think the discovery of the phosphodietate by Fritz Eckstein, that was definitely an enabler because it allowed us the bioavailability. And I think without that, the current oligonucleotide would not be there. Other things like manufacturing, scaling, all these things are crucial and they may seem like the more boring part, and all the chemical modifications. You can make a chemical modification, but if you can't scale up, then it will never become a therapy. So people that did work on that, I think there's also definitely a role there. And I think what is nice, and well, disclaimer, I've been the president of the Oligonucleotide Therapeutic Society, so I'm probably biased, but the Oligonucleotide Therapeutic Society really allowed for a dialogue from an early stage, very open dialogue, research-driven, data-driven, not just from academia, but also from industry. And I think that helped as well to move the field forward, to have this from an early stage.
Thank you very much, Mika. I think you mentioned a couple of topics as well, which we will definitely further discuss in the CMC section, which will follow this session. Tal, can I ask you as well about your view about maybe the major advantage we recently discovered?
Well, I think there's been a wonderful congruence in the field in the sense that, let's not forget, we probably wouldn't be able to do all of this if it weren't for the availability and cost of sequencing having come down. Right. At the end of the day, we're talking about a modality that is driven by the sequence as the fundamental architecture of everything that we do. So I think the ability to integrate sequencing into everything, even quality control, right, let alone looking at patients and diseases and understanding mutations and coding and non-coding, has really played a critical role here.
Thank you, Sol.
No, full agreement with Tal. Yeah, and also, as Mika said, obviously, yeah, it seems that things came up out of nowhere, but it's true that there's so much research in the field that we can build from all of that. So, and again, from the regulatory perspective, I think we will take all those messages regarding quality requirements that obviously will depend on the type of molecule and the mechanism of action. But in agreement with Tal.
I do want to add one other point, if I may. As I think about this, and it's exemplified by this conference, I think COVID also put a different level of scientific collaboration at the forefront of everybody. The ability to share scientific data, even in preprint form, and yes, maybe some of it turned out to be wrong, but I think we will all agree that the overarching benefit of having early access and open access to data and to dialogue that we all experienced during COVID, I think has been a really important enabler of that success. And I hope some of those elements stay with us as we consider how can we all do better, even within our respective niches of where we operate, to further advance these medicines and make them available faster.
Sol?
Sorry if I may, didn't explain much because I was running out of time through the last slide. Just to mention, yes, fully agree that good collaboration, obviously, it's going to be very helpful. And there is this open initiative that we will move forward, from the EMA to open the regulatory process and discussions to other regulatory authorities. We have now a very good well-established collaboration with FDA, with other agencies, but this project will move forward and tries to open, deliberations and discussions to other agencies. So I think that would be very useful. So looking forward to that project. Thanks.
Thank you. This nicely reflects as well the opening remarks from Ima, our director, that coming out of this pandemic with all this negativity, that we should try to harvest really some advances we made there. Thank you very much. One more question, one more general question. What about failures? We all know about the bias of publishing negative studies. What did we learn from failures, or what can we learn from failures in this area? And maybe Mika first.
No, I can definitely start. And I think the first thing, what Tol also already mentioned, we need to publish. We need to publish what works, what doesn't work. And also, if we're not really clear, we see something strange, rather than taking 6 years to elucidate everything and having other people maybe working on the same thing, it's good to publish early and to share things early. And I think also one thing that we learned as an oligonucleotide society is it's crucial to take along proper controls. So those who are new to the field may not know this, but say 20, 25 years ago, the oligonucleotides got a lot of bad rep because people were using them as an anti-cancer drugs, but then oligonucleotides can also cause inflammatory effects. So they said they had sequence-specific effects, but actually what we were looking for was just a generalized inflammatory response that was not sequence-specific. And as soon as you take along the proper controls, you can elucidate these and you can know, well, is this targeted or not? But it took a while, and I think this is the danger. If people do sloppy science, then it's easy to get wrong conclusions and give a whole field or a whole area a bad name. So I think that's one thing that we learned as well. It's crucial to take along proper controls. And I think a last thing that we learned, and that's more from the Duchenne field that I'm part of, is that drug development with oligonucleotides is not just the oligonucleotide. You don't only need an oligonucleotide that is effective. You also need other infrastructure. You need outcome measures. You need things for trial design. And you need to know how regulators approve drugs. And as the Duchenne field, we didn't know that. When we started our clinical trials, we were clueless. So we started our interaction with the regulators too late. And I think that's a warning and a lesson that I share here for the academic viewers. If you're developing a therapy, start the dialogue with the regulators early, because you need to anticipate on things. And if you need to develop new outcome measures when you have your compound ready for clinical trials, it's too late.
Thank you very much, Mika. Tal, with your rich experience in development, very successful ones, but maybe some failures as well. What did we learn? What can we learn from that?
Well, I think in our history, it's interesting. The biggest failure we had was when we realized in one of our vaccines that we got the sequence wrong. And for me, the beauty there was that within less than a year, we simply fixed the sequence and we were back in the clinic. And so, the attention to detail here, I think the other element that the field is struggling with, frankly, is the preclinical translatability of model systems as it relates to this. And I think here there have been numerous challenges by many along the way. The old saying goes that, mice lie, and mice don't lie. Mice, for some things, are relevant species, for others, they are not. And I think we really need to find better ways to have more predictive preclinical models without over-relying on NHPs to make sure that when we get into clinical experiments, what we're doing is right. And here, I would hope that the totality of experience that we have and somehow the body of knowledge will enable us to do less than NHPs, leverage more of platform approaches and expectations, especially as they relate to delivery, and come up with science-based but more effective ways of predicting human clinical benefit risk than we currently have. I think that is still a challenge that many of the companies that I'm involved with are facing on a daily basis.
Thank you very much, Tal. Sol, from the regulatory perspective, I don't know, maybe over-regulation, under-regulation, do we need more guidance? Do we have too much guidance? What's your experience?
No, thanks. No, fully agree with the messages from Mika and Tal. Obviously, I think we need guidance in this particular area, maybe also, specifically more on quality and non-clinical, but full agreement. I think we need to learn, and we talk a lot about platform approach. I think, in these areas and the limitations that we are seeing, translating the information from non-clinical studies to humans. So we are already seeing that with some ATMPs. So I think, we have to build from experience and move forward. And if we can be more efficient with maybe, less studies on the clinical side and build more on the knowledge that we have from other platforms, similar platforms, obviously that is going to be efficient. And I think from our perspective, I think, we are also, we can also learn from failures. In the sense that, the system or the regulatory system has become more transparent, more and more with time and increasing. So it's important that, for instance, if a product doesn't make it or if it gets a negative opinion, that the information is out there, with the limitations, obviously, of certain confidential information. But I think if this is published, and also obviously the positives that get to the market, and we provide the rationale why there was considered that there was a positive benefit risk, I think this is very helpful for other developments. Thanks.
Thank you very much. I'll now go into some questions received online. Is platform or prior knowledge something that can be applied to other RNA modalities, such as siRNAs, ASOs, etc., as well as it is done for mRNA vaccines? And what would define platform knowledge? We have platform as a topic in several other sessions, but I think to have a start here, who would like to make a start? Mika?
I'm happy to make a start. I think so. The thing with platform, if you do, so I think we have to be careful. So what is a platform? I think what we're not looking for is a blanket approval of all siRNAs or all mRNAs, but we're saying, can things be streamlined based on the knowledge that we have and within each modality? So I think that's definitely, we can't transfer all the knowledge across modalities. So we really have to look modality specific as RNA, RNase, etc. But definitely we can learn. So based on the information that we have from other compounds for the same, using the same modality, we can learn from what we know about PK, whatever. But I think we also have to realize that it's early days yet. because each of the different modalities, there's only a few compounds approved yet, and there's a lot more in the pipeline. But I think there's maybe not enough to now confidently say, well, we know the safety profile of the next 100 oligonucleotides that will be developed for brain diseases, for instance. So I think we have to keep collecting data, and hopefully there will come a point that we can be talking about these platform approaches. I think also we have to look carefully when we develop these things, like which are things that make sense and which are things that don't make sense. So for each modalities, there's probably parts of the development currently that really make no sense and that are a waste of animals. I think there we can have a critical look. But I would say it's too early yet to just talk about modality or talk about platforms because we have very limited information yet.
Thank you, Mika. Tal, please.
Yes, I actually want to thank the regulators here for often being the source of collaboration and inspiration to some of the drug developers, because you are aware of potential shared, especially safety risks, sometimes before we are. And there are several examples recently, I think even in the Duchenne field, of figuring out adverse events across different sponsors who got together when a shared signal was seen. And I think that's then for the benefit of everybody who comes later. So I do think there are emerging commonalities that are seen, particularly as it relates to expectations for adverse events and what to monitor in the clinic. And I think the regulators are already playing a critical role there. I think beyond that, as I'm sure will be discussed in the following session, there are critical quality attributes that are shared by platforms, but by the nature of the definition are going to be restricted to that manufacturer. And those are going to be, I think, very hard. It's easier for me in the clinical realm to say, okay, thrombocytopenia with ASOs, yep, been seen before, got to go check for it. I think that's much more difficult to implement in areas of CMC and quality control. From a platform perspective, they become very manufacturer specific. Over.
Thank you, Tal. Sol, you heard it. You are the enabler. How do you do that?
Yeah. Thank you. Thanks so much. I think, I'm in full agreement with the comments from Mika and Tal again. What is a platform? That is the million-dollar question that we haven't solved yet, even for more established medicinal products such as monoclonal antibodies. I mean, we talk about platforms, and obviously, we have many monoclonal antibodies marketed, and obviously some quality requirements and production process and controls are very similar, particularly within the same company. So I think I agree that it's maybe too early to consider platforms on RNA-based medicines. Mika really commented very well. But I think maybe we can start with little steps, and I can see how, for instance, from the same company, if we have a particular mRNA vaccine, and now we have a different antigen, but it's similar in the structure, similar in the mechanism of action, similar in production. Well, there's already some basis to learn from that. So I think, again, maybe little steps, but we can move from there. Thank you. Thanks.
Thank you very much, Sol. I have one question for Tal. Providing an individual cancer vaccine for every individual patient, how should we address efficacy, quality, and safety of personalized mRNA vaccines and other personalized medicines without skyrocketing costs for the treatments? Tal, how do we do that in the future?
Yeah. How much time do we have?
4 minutes.
This is okay. Then it's easy. Here's the deal. Science and technology can actually enable a lot more than we often can move effectively and efficiently in the clinical development. And the cancer vaccine for me is a wonderful example. There's no question it's a platform. Call a platform what you will, but that's it, because it's a different drug for every patient. I think technology has enabled it to be in the realm of accessibility. It's not going to be cheap, but if the effectiveness in phase 3 holds to what we've seen in phase 2, it's going to be definitely worth it. And I'm sure that with scaling up, I mean, the beauty of these technologies is that they're actually cheaper in some instances than what we've gotten used to with biologics, right? And so I think with scale up, the company should be able to bring the costs to a place where it's clearly worth the value. The challenge, I think, is going to be, to your point on quality, I'm less concerned because I think there are, it's becoming fairly mature fairly rapidly. What are the critical quality attributes there? And I think the company should have a good handle on it. It is really the efficacy, from the safety as well. It's the vaccine. Recall that in a cancer vaccine, we give 10 times as much as we did in a COVID vaccine. We do it every 3 weeks and it seems to be fine. So I'm less worried about that. The efficacy is an interesting challenge because the efficacy is a function of how well the algorithms predict for a population the binding from which the vaccine actually chooses the antigen. So, we all know in immunology that the most common haplotype is the one where we have the most data. Well, software has got to improve over time so that that prediction is as good for people who are more diverse and have less common genetic background. And I think that's where the challenge is going to be. How do we ensure that the software that was developed within a certain population to predict what is going to bind, actually, when we move it to a different population, will still perform? That's not a trivial problem at all. And I don't think there's an easy answer. I would submit that the obviously wrong answer is to expect to run different phase 3 trials in different populations every time we have, a different set of software. I think we're going to have to come up with a framework there that's novel, that enables us to somehow make judgment on the suitability of the software to the population we care about, and then develop the systems in real world evidence once people have gotten it, to ensure that indeed we're seeing the benefit in the real world that we expect to see. I can't think of a different way to solve this. Other than that, but it does provide a fascinating edge case, if you will, for any conceptual framework we have. Because if we solve that rare disease for a different mutation in the same gene, giving the same phenotype and corrected the same way, that's going to kind of look simple relative to that.
Thank you very much, Tal. Okay, I think to the last question, all three of you, very brief response, please. If you have a magic wand and you have one wish, what would you like to change in the healthcare system, in the development of RNA-based medicines? Very brief, one wish. Tal, then Anmike, and then Sol.
What I would like to change is, if I had a magic wand, it's the public perception of what we do and the distrust that, even despite the COVID vaccines, is still out there, and the implication it has for the public's willingness to share the critical health data, without which we struggle to, frankly, develop medicines. And I think it's true for all medicine, especially true for RNA-based medicines that are so heavily dependent on understanding genetic makeups. I think the level of distrust and fear for privacy there far exceeds what I think is a reasonable risk, and that's, I think, a real challenge for the field.
Thank you. Anmike?
Yeah, so not really the regulatory system, but I have a magic wand, so I can change the question. I think so for the rare diseases, I think we're collaborating a lot pre-clinically. We're collaborating as regulators, and there's the pan-European approach to approve rare disease treatments. But then the marketing and evaluation is done country by country. And I think, well, if I had a magic wand, that's what I would change. I would say, let's do this European-wide rather than country by country, because the inequity now where you have drugs that are available in one country for rare disease patients and not in others, I think that's just, well, that's not how it should be. And I understand that people in this room and in this meeting probably can't do anything about that, but that would be more my wish than changing the regulatory system.
Thank you very much, and we certainly take this comment into consideration. Sol, please.
Yeah, no, I think obviously, I mean, good communication. I mean, it's critical. So it would be good if we could, I don't know, continue learning and improving our communication. Sometimes, you know, regulators, I think, have a different task, which is how to put things in the proper balance, you know, the benefits versus the risks, and how to communicate this in a proper way, because that's what we do, and nothing is perfect. And as Mika said, I think probably, you know, increased dialogue among regulatory agencies obviously would really benefit the system. I understand that different regions have different regulatory frameworks. Sometimes it's difficult, but at least, you know, having a dialogue on scientific issues, what are the concerns on quality that could translate to efficacy and safety, and maybe, you know, have a more common approach. I think that would be obviously very helpful. Thank you. Thanks.
Thank you very much, all three of you. Very nice and lively discussion. And I'd like to hand over back to our co-chair, Steffen.
Thank you, Falk, and thank you both to Tal, Mika, and Sol for this very interesting presentations and discussions. Now it's time to adjust your caffeine serum levels and maybe have a short bio break. So see you back here at 11 CET. Thank you. Dear all, welcome back. I hope you had a quick break, got adjusted your caffeine levels and what else needed to be done in 15 minutes. We are back here to kick off our next session on CMC for RNA technologies. It's chaired my colleague, Brian Dooley. So I pass on to you, Brian, please.
Thanks, Stefan. Good morning, everyone. My name is Brian Dooley from the Pharmaceutical Quality Office here at EMA. And I'm excited to be moderating this session 2, which is on the application of RNA technologies in CMC, chemistry, manufacturing and controls. We have 1 regulator and 2 industry speakers who will give their presentations, and we'll follow that with a panel discussion where we'll have another 3 panelists who I'll introduce for the Q&A. So our first speaker is Dr. René Thurmer from BfArM, the German medicines regulator. René received his diploma in chemistry and PhD in biochemistry from the University of Tübingen. He joined the BfArM in 2000. He currently serves as a CMC reviewer and is the deputy head of unit of pharmaceutical biotechnology. His experience is in the field of formulation, manufacture and control of medicinal products, in particular in the field of oligonucleotides, peptides, proteins, liposomes, sustained release polymer drug products, depot formulations, conjugated drug products, natural and synthetic surfactants, and nanomedicines. He is currently a member of the EMA's Quality Innovation Group. So with that, I give the floor to you, René.
Okay, thank you much, Brian, for this nice introduction. So next slide, please. So this typically a disclaimer. So next one, please. So this slide provides you with an overview of all marketing authorization applications received for synthetic oligonucleotides. And on the next slide, you will see an overview of review issues from the last 5 years. These were 9 procedures, and you can see there are 2 major fields where a lot of questions were raised from the regulators in Europe. The first one is the manufacturing process. That means description of the manufacturing process, control of material, control of critical steps and intermediates. And the second one, the big field, is the control of the substance or control of drug product. Everything here is related with specifications, analytical methods, method validation, and so on. Next slide. From the experience we gained during the review process from all these applications, we decided that it's time to draft a guideline on the development and manufacture of synthetic oligonucleotides. So the concept paper was published in September last year, followed by a 3-month consultation time. And on the next slide, I will provide you with some information and a summary what's written in this concept paper. So it's stated that synthetic oligonucleotides are at the interface of small molecules and biologicals, and from a quality point of view, specific considerations apply to this class of therapeutics. And the guideline will cover antisense and other single-strand products, double-strand products as siRNA, and third subclass, the aptamers. So proposed guideline will address the following. First, the development of an overall control strategy to ensure consistent quality of synthetic oligonucleotides and the resulting medicinal products based on relevant CQAs. Then the requirements specific for the solid phase synthesis manufacturing pathway, including requirements on batch definition and the description of splitting, pooling, and reprocessing steps applied in a purification process. Then the next point is the selection of starting materials, including a criticality assessment of their impurity profiles. Then the characterization approaches, including investigation of the drug substance impurity profile. Next slide, please. Then the purity control strategy will be addressed. That means product-related impurities and process-related impurities, and the use of orthogonal or complementary purity methods, and a discussion of grouping strategies for these impurities. So difference in the requirements between the single-strand, double-strand, and aptamer oligonucleotide products will clearly be outlined. And what is also important and has been addressed in the previous Q&A session, so oligonucleotides definitively have a potential to use prior knowledge and platform technology, and we will try to give some recommendations on how to justify the applicability of prior knowledge and platform technologies in this guideline. Also, an increasing trend to use some conjugations is observed, so we will also address the requirements for conjugation, for example, for GalNAc conjugates, pegylation, and also a relatively new trend, the conjugation to monoclonal antibodies, peptides, and proteins. So the guideline will follow the structure of Module 3 and the European guideline on chemistry of active substances where relevant, but also we will address some finished product considerations, for example, in choice of excipient, formulation, and also at the moment very difficult or controversial point, the sterilization aspects. This will be also addressed in some of the next presentations. So another important topic is the purity limits, if we need purity limits, if we do not need purity limits. So in contrast to synthetic peptides, we have no purity limits in the EDQM Pharm.io monograph for substance of pharmaceutical use. We have these limits for synthetic peptides, and establishing harmonizing limits could be considered in this guideline. So, but additional discussions are needed as the analytical methods and specific oligonucleotide type of products are not comparable per se. So we stated in this concept paper that the public consultation may facilitate comments from different stakeholders in this regard. The next topic of the guideline will be considerations for active substance that are in your solution. This is also a relatively new concept, and also in the future we expect the first submissions for generic oligonucleotides, and also here we need additional considerations regarding quality aspects may be applicable for such submissions, including demonstration of comparability. And also we have initiatives for the development of personalized antisense oligonucleotides, either for one patient or a group of small patients, a small group of patients, sorry, and this is also will be addressed in the guideline. We have received 61 comments from 7 stakeholders, And the majority of the comments are regarding the purity control strategy and specifications. Of course, also a lot of general comments, classification comments, comments on analytical methodology. And on the next slide, please. I tried to give an extract of some key messages from this public consultation. The first one, that industry wants a clear definition of the scope. That means which molecule classes are within or without the scope. Then international harmonization would be appreciated by industry, and no surprise, no additional regulatory burden. The EPOC, this is an industry consortium for oligonucleotides, has published several white papers in the past, and industry also wants that these these white papers are considered in the drafting process. One question is also how to address mutagenic impurities, because synthetic oligonucleotides are formally excluded from ICH-M7. Then I think the most relevant part will be the discussion on requirements for specifications. We received several comments on analytical techniques, and also industry requests clarity on the expectations for benefit risk analysis for sterilization method selection. That means sterile filtration versus the terminal sterilization. We were discouraged from trying to establish generic purity limits because these are too dependent on the chemistry sequence, even within the same chemistry and method. So I think this will be also a potential topic for the Q&A part of this session. And also, but this is also in our interest, phase-appropriate approaches to CMC development during clinical development. Next slide. So some of our experience from the last years looking through the different dossiers, for example, for manufacturing process development, the majority of the dossiers is in line with the expectations from ICH Q11 and Q8. And these sections in Module 3 are usually of good quality. So design of experiment studies are usually performed in small scale to optimize the manufacturing process. There's an identification of the normal operating ranges, set points, and approval acceptable ranges, and also criticality assessment of process parameters and identification of CPPs for synthesis, purification, and annealing in the case of double-stranded oligonucleotides. Then the criticality assessment of quality attributes and justification how these attributes are controlled. And finally, a definition, description, and justification of the overall control strategy. Next slide, please. So starting materials are one of the most important parts of the overall control strategy. We have unique challenges for oligonucleotides, and the phosphoramidites used as starting materials are more complex than many small molecule APIs, but they're generally accepted as starting materials. We have more complex sequencing in development programs. So there's a complex chemistry and synthesis for starting materials that should be considered. And also we have many development programs with linkers and conjugates, and there are also several issues with this regard. What's important is that the justification for the designation of the starting material should be provided in line with ICH Q11 and the questions and answer document for this guideline. So also documentation of suppliers is expected. This is different to other small molecules. For example, we have a huge number of starting materials with a huge... Sorry, could you please go to the previous slide? Thanks. No. Thank you. So a huge number of suppliers, and this should be documented, and also a short description of the starting material manufacturing process should be provided. And the criticality assessment of the impurity profile is essential for oligonucleotides. And also the impurity profiles for different suppliers and the phosphamidite should be compared. Next one. So impurities is also very specific for oligonucleotides because we have a lot of co-eluding impurities and groups of impurities. So, but our observation is the good understanding of impurity profiles has been demonstrated, and for process-related impurities, the depletion should be demonstrated. So a risk analysis and theoretical purging considerations may be a suitable approach. However, also some analytical data are requested for specific impurities. Genotoxic impurities need to be discussed in the dossier. There's a very recent publication also from the EPOC. Nitrosamine is at the moment in the focus. Questions and answers are published on the EMA website. And also for starting material impurities, understanding of their criticality on the final IPA quality should be demonstrated. And also aggregation could be an issue for oligonucleotides. So for the specifications, we have unique challenges for oligonucleotide product-related impurities. So high analytical methods are a prerequisite, and the grouping strategy has to be justified by the applicant, and the justification of specification section is extremely important. Key is the toxicological and clinical qualification, and also some comparability studies during the development using high quality analytical methods. The manufacturing process validation should show that the commercial process results in a product that falls within the clinically qualified limits, and usually the plus-minus threefold standard deviation approach is adequate for setting of specifications. But also data are sometimes limited, so post-approval adjustment of the specification may be an option with some post-authorization measures. So early development batches should not be the basis on specification setting per se, and differences between the release and shelf life specification for the drug product should also be justified by data and knowledge of the degradation pathways. So next slide, please. So what are regulatory trends? Usually for antisense and siRNA molecules, no bioassay expected. We need it for aptamers. However, the justification for the omission should be provided, and also the consistency of the distribution of stereoisomers should be evaluated, and not including a routine test on stereochemistry should be justified. We need process validation data for drug products sterilized by non-standard procedures, and it's the thinking of the EMA or European regulatory assessors that the terminal sterilization provides the highest assurance of sterility. We have a guideline on this, including some decision trees, and this guideline is definitively relevant for synthetic oligonucleotides. We have received numerous scientific advice applications regarding this topic, and unfortunately in numerous cases the justification and the provided data package is not considered sufficient. This will be also ongoing discussion. Next one. So what are typical questions for scientific advices? We received 11 scientific advice procedures with 51 quality questions last year, 6 procedures with 23 questions in 2021, and there are a lot of questions regarding bridging approaches during clinical development. specifications for active substance and finished product, starting materials, analytical testing, process validation strategies, manufacturing stability programs were often reflected in the briefing books, and also the sterilization issue. Next one. So at the end of this presentation, of course, we have a clear focus on synthetic oligonucleotides because we are now in the drafting process for the guideline. But now to the mRNA therapeutics. We have limited experience for mRNA therapeutics. However, significant regulatory quality knowledge has been gained from the COVID-19 mRNA vaccine applications. And from my point of view, the quality considerations for therapeutics and vaccines are very similar. And platform approaches are very, very likely and makes definitively sense. So important are the comparability considerations during development, and that the consistency and robustness of the manufacturing process will be demonstrated. The quality of materials and excipients is extremely relevant, and also the qualification of multiple suppliers, for example, for the lipids. We have also some observations, interesting data regarding the characterization and the extended characterization. This also has been to put in balance with the routine testing and stability testing, more information for the characterization and less routine tests. So then stability, storage, and transportation are of course an issue. And also here for the mRNA therapeutics, we have received a number of scientific applications, which is steadily increasing for a diverse type of products. And the main recurring questions in scientific advice procedures are PPQ strategies, potency testing, analytical methods, and comparability. And also we have the first prime designations for mRNA therapeutics. Next one. So what are the typical attributes which should be considered? Of course, the compendial methods, experience, pH, and so on. Then identification, of course, assay and content, integrity, purity, the sequence, potency is very important. This will be addressed in one of the next presentations. Capping efficiency, residual DNA template, also residual double-strand RNA, and of course the delivery system testing. That means, for example, size distribution, encapsulation, and release of the mRNA. Also some lipid-related tests, identity, content of the lipids in the lipid nanoparticles, and also impurities resulting from lipid degradation. So next one. This is my last slide. So I am happy to announce that the work plan of the Biologicals Working Party at the EMA for this year includes a proposal for a draft EMA guideline on the quality of mRNA vaccines. So a concept paper on this topic for public consultation is expected by mid-2023. We also have to consider guidances from other regions or organizations, for example, from the HAW or from OSP. And also the EDQM has recently established a new working party for mRNA vaccines, and the first meeting, face-to-face meeting of this group will be next week. And with this, I would like to give back to Brian.
Thank you, Rene, for a very comprehensive presentation. I can see already some questions coming through on the Slido, so a reminder to participants to please take a look at the questions. You can enter your own questions, and you can like questions of others, which will put them higher on the list for us when we're selecting for the Q&A. Our next speaker is Daniel Capaldi, who is Vice President of Analytical and Process Development at Ionis Pharmaceuticals. Daniel, we know you're in California, so it must be the middle of the night there. So we want to say thank you very much to you, or the very early morning at least, for joining us today. Daniel received a PhD in chemistry from King's College London, and he's been with Ionis since 1996. In his current position as Vice President, he contributes to the development of antisense therapeutics and has responsibility for a variety of process chemistry and CMC functions, including supply chain and process optimization, analytical method development, release and stability testing, and impurity characterization. He was lead author on 4 marketing applications for approved oligonucleotide therapeutics, so Canamro, Spinraza, Tegsedi, Waylivra, and he's the author of over 30 papers and patents in the field of oligonucleotide chemistry. So, Daniel, thank you for joining us this morning, and the floor is yours.
Thanks, Brian. Great. Okay, today I'm going to present the European Pharma Oligonucleotide Consortium's perspective on some CMC topics relevant to the commercialization of synthetic oligonucleotide therapeutics. Next slide, please. Here's the agenda, but let's just skip over that quick and go to the next slide. I wanted to start by giving a very brief introduction to the European Pharma Oligonucleotide Consortium, or EPOC. EPOC comprises a group of innovator companies actively engaged in the development of oligonucleotide therapeutics. You can see the current members on this slide. EPOC's mission is to identify areas for harmonization of oligonucleotide CMC development and propose solutions with the intention of expediting access to new and important medicines. In practice, what this means is that we intend to collaborate to produce a series of white papers that we hope will shape how therapeutic oligonucleotides are manufactured, tested, and regulated, and which generally help to move the field forward. Next slide. I thought it might be helpful to provide a list of the papers that we've published so far. I've also included on this slide another couple of papers that were published ahead of the formation of EPOC, but which seem to have gained some traction. Obviously, in the time available today, I can't hope to cover all of these topics, and so I've decided to speak on just 3. I chose the topics because, judging from the comments that I've seen on the EMA guidance outline, these seem to be areas that people are interested in. And in addition, each seems to me to contain aspects that are particular to oligonucleotides. Next slide, please. So, drug substance starting materials. This is subject of a paper recently published in Nucleic Acid Therapeutics. The goal of the paper is to create a harmonized approach to justifying the selection of regulatory starting materials. The focus is phosphoramidite starting materials for solid phase synthesis. The authors recognize the value of Q11 and suggest applying as much of that guidance as possible. So, for example, starting materials for oligonucleotide therapeutics, like all starting materials, should have defined and stable structures and characteristic, chemical and physical properties. And there should be the requirement to provide some proof of structure and characterization data. Next slide, please. It also seems vitally important to characterize and determine the fates of impurities in starting materials. And here it may be useful to assign impurities as critical or non-critical. Actually, it turns out that the major impurities in nucleoside phosphoramidites are unreactive. And in solid phase synthesis, I think it's reasonable to assume that non-reactive impurities simply flow through the column to waste and therefore can be considered non-critical. Further, in most cases, non-reactivity should be fairly obvious, and so I think there ought not to be a requirement to conduct formal fate and purging studies for such impurities. On the other hand, reactive impurities are likely more troublesome in solid phase synthesis than in typical small molecule syntheses. First, there's no purification between cycles, and therefore propinquity arguments are essentially meaningless. Second, starting material derived product-related impurities are typically structurally similar to the parent and so hard to remove by purification. And third, there's this potential for these multiplicative effects, which means that even very low levels of starting material impurities can lead to appreciable amounts of drug substance impurities. Next slide, please. Naturally, starting material specifications should be focused on testing starting material critical quality attributes. And really, there are only two attributes that are critical: starting material identity, which of course determines the identity of the drug substance, and the amounts of reactive nucleoside phosphoramidite impurities, which will likely need to be controlled to low levels. Despite this, it's not uncommon to be asked to add a whole raft of additional tests that don't measure anything important. And I've listed some examples on this slide. In my view, it would be better to focus efforts on getting the critical measures right without getting distracted by a long list of meaningless tests. Next slide, please. Moving now to drug substance and drug product impurities, which are covered in the paper linked here. The focus of the paper is product-related impurities, which come in these three flavors here. The paper endeavors to cover chemistry aspects, such as impurity characterization and reporting, and safety aspects, such as impurity qualification requirements. Next slide, please. Impurity characterization. We can probably agree that it's important to make some effort to characterize drug substance impurities, at least by the time of submission of the marketing application. Of course, characterizing impurities in a molecule as large and complex as an oligonucleotide isn't trivial, and therefore I think it's reasonable to ask how much effort ought to be expended. And maybe there are no hard and fast answers, but it seems to make sense that there might be a spectrum of empirical data requirements, with very little required for impurities previously characterized by several groups for several oligonucleotides, for example, the PO impurity of a phosphorothioate oligonucleotide, and much more required for impurities that are observed for the first time. Next slide, please. Impurity reporting. We can probably agree that the specification, the drug substance specification, should include tests for a list of specified impurities, which we might define as those that are observed frequently and that are diagnostic of the success or otherwise of a particular step or control strategy in the manufacturing process. For example, single-stranded oligonucleotides typically contain small amounts of N-1 impurity, the levels of which are largely dependent on completion of the detritylation step. Therefore, monitoring N-1 in the drug substance may help confirm that the detritylation step performed as expected. Another aspect that comes up frequently and was just mentioned by René is that of impurity grouping, as it's generally the case that complete separation of impurities isn't practical. I guess there are several ways you might group impurities, but one way is by structural class. For example, you may think to report N-1, N+1, and abasic impurities as separate groups. I think this is appealing because all of the members of the group are subject to the same mechanism of formation. And therefore, grouping by structural class is consistent with this idea that the reported results confirm the success of a particular step or control strategy. Next slide, please. What about the reporting threshold, or the level above which there's an expectation to report in some impurities? For most small molecule drugs, the reporting threshold is 0.05%. I think the important consideration here is the LOQ of the analytical method, as it's recognized there isn't much point reporting impurities that are present below the LOQ. As impurities of small molecule drugs are typically single components readily separated chromatographically as sharp, well-defined peaks detected by UV absorbance, LOQ values on the order of maybe a couple of hundredths of a percent are readily achievable, and therefore a reporting threshold of 0.05% is feasible. On the other hand, product-related impurities of oligonucleotides are often difficult to separate chromatographically, and in some instances can only be detected by mass spectrometry. And when chromatographic separation is practical, the composite nature of impurities contributes to peak broadening, which obviously reduces signal to noise and impacts the LOQ. In addition, mass spectrometry detection typically has higher LOQ values than UV detection. However, I think it's also the case that oligonucleotides often contain a greater number of impurities than most small molecule drugs, and therefore, if the reporting threshold is too high, we risk severely under-reporting total impurities. So maybe a reasonable compromise is 0.2%. Next slide. As I said, the paper also covers safety aspects of product-related impurities, and specifically when and how one might want to qualify impurities. One of the main ideas is that qualification requirements might be based on impurity type. For example, impurities that are also major metabolites, impurities that contain only structural elements found in naturally occurring nucleic acids, and perhaps a little more controversially, impurities that are sequence variants of the parent might not need to undergo tox qualification, which would leave only those impurities that contain structural elements not found in the parent or in nature. Next slide, please. Furthermore, for these types of impurities, which we classify as class 4 in the paper, perhaps it would be acceptable to use tox qualification data from one oligonucleotide to support qualification of the same impurity in a related oligonucleotide. I think such a concept would be aligned with database structural alert approaches such as tox alerts and Derek, and it would also be consistent with the sentiments underlying the recently enacted FDA Modernization Act 2.0. Next slide. Well, so much for specified impurities, but there's probably also the need to include a test and limit on unspecified impurities, which by standard practice is set equal to something called the identification threshold. I think this is driven primarily by quality expectations. That is, there's the general quality expectation that formation of new and unidentified impurities above some threshold be avoided. And further, that when operating in a state of control, validated manufacturing processes and control strategies should be capable of rendering drug substance or drug product that contains less than the identification quantity of a new impurity. For most small molecule drugs, the identification threshold is set at 0.10%. I believe the same quality expectation should apply to oligonucleotides, but if the identification threshold is driven largely by what we should be capable of doing when everything works as planned, I think we need to consider the following. Oligonucleotides are typically produced by solid phase synthesis, where the products of one reaction are fed into the next without purification, and so there's little opportunity for impurity removal. Minute quantities of reactive impurities in starting materials may lead to significant quantities of drug substance impurities. This is this multiplicity effect. Oligonucleotides present complex impurity profiles, and it's often difficult to distinguish impurities from the parent and from each other. Of course, all of this obviously places practical limitations on our ability to control unspecified impurities, even when everything is operating under a state of control. And therefore, the small molecule identification threshold of 0.10% needs adjusting up. And we suggest in this paper that 1% is about the best we can do for most oligonucleotides. Next slide. Okay, but is 1% of a new unidentified impurity okay from a safety perspective? Here, I think the answer is yes, for the following 4 reasons. First, unspecified impurities likely comprise the parent oligonucleotide modified at a single internucleotide linkage, sugar, or heterocycle. Therefore, the physicochemical properties of the impurity are very likely similar to those of the parent. That is, they're both water-soluble polyanions of similar molecular weight and protein binding properties. 2, there's a large molecular weight difference between oligonucleotide and small molecule impurities, and therefore the same quantities of each represent very different molar amounts. At the same clinical dose, 1% of an oligonucleotide impurity is approximately equivalent in molar amounts to 0.1% of a small molecule impurity. Third, most oligonucleotides are dosed at much less than 100 milligrams a day. Therefore, 1% of the daily dose is usually much less than the 1 milligram amount that's often discussed as generally being safe for all compounds. And there's a reference to that here. Finally, I think there's just the availability of tox data for common impurities of oligonucleotides. And I'm not aware of any toxicities attributable to an impurity. And so because all impurities that we've tested to date are well tolerated, I think it's likely that future impurities are also likely to be well tolerated. Next slide, please. Okay, the last topic I want to present on is, again, something René mentioned. It's terminal sterilization, which is covered in an EPOC paper that should be available very soon, and which I genuinely believe will be required reading for anyone interested in oligonucleotide drug development. The goal of the paper is to fill gaps between regulatory expectations and the application of terminal sterilization, or TS, to oligonucleotide drug products, with special attention to the recently published EMA guideline, which is linked here. Without doubt, this is the most useful but prescriptive guideline on TS published. The document provides guidance on the selection of methods and technical requirements while making it very clear that efforts should be made to enable terminal sterilization. We're also told that increases in impurities above the qualification threshold does not preclude the use of TS, and really left in no doubt that TS is the preferred option, and therefore the use of sterile filtration and aseptic processing must be justified. Next slide, please. The current situation is that it's generally accepted that proteins and other biologics are too labile to withstand TS. And so far, all oligonucleotide drugs have been manufactured by sterile filtration and aseptic processing. The authors of this paper enumerate several key concepts, starting with a theoretical risk assessment. Here, the main conclusions are that double-stranded oligonucleotides with TM values below 110 degrees, aptamers, and oligonucleotides conjugated to proteins are likely excludable from the outset. But that, however, some single-stranded oligonucleotides are more thermally stable and are therefore hard to exclude without further work. For oligonucleotides that can't be excluded by theoretical risk assessment, advice on formulation development is provided. For example, phosphate is recommended as the most suitable buffer on the basis of its buffering range, negligible temperature coefficient, and general compatibility with oligonucleotides. Next slide, please. There's also an in-depth and immensely helpful discussion on the development of the sterilization cycle itself, starting from the EMA requirements on indicators and accepted limits on initial bioburden concentration. The authors argue that a cycle lethality of F sub 0 of greater than or equal to 12 minutes is required to achieve a sterility assurance level of 10 to the minus 6. And therefore, you should target F sub 0 equals 12 during feasibility studies and not the minimum F sub 0 of 8 minutes referenced in the guideline. We're reminded that degradation occurs during temperature ramping when the temperature is less than 110 degrees C and there's no additional accumulation of cycle lethality, and that the goal of the development studies is really to mimic the anticipated capability of the commercial equipment. It's also the case that autoclave cycles are commonly controlled by setting the hold temperature and time, and therefore we should include tolerances expected in commercial equipment. For example, a temperature band of 2 degrees is typical, and therefore the authors recommend using a temperature at the top of the range. We're reminded that there's really no sense examining lower temperatures for longer times, as decreasing the sterilization temperature by 10 degrees results in 10 times increase in time to achieve the same lethality, whereas the rate constant for most chemical reactions decreases by a much smaller fraction. The authors admit that higher temperatures should lead to less degradation, but of course there are some practical limitations in commercial equipment. Next slide, please. The main conclusions from the paper are as follows. For aqueous products, you should assess moist heat sterilization methods per the EMA decision tree. Irradiation and chemical methods are not recommended, and they also don't recommend partial heat treatments. Feasibility studies should be carried out using the most heat-stable formulation available and container closures of representative materials of construction. And the goal of the studies is to generate sufficient data to rule confidently on the feasibility of terminal sterilization. terminal sterilization is not recommended when any of the following occur. An impurity increases beyond its toxicological qualification limit, which is consistent with the EMA guideline. Significant changes occur to other critical quality attributes, for example, appearance or visible and subvisible particles, pH. Total degradation is sufficient to risk failing the lower limit on assay, or chemical or physical changes to the container negatively impact its suitability or functionality. And next slide. That is the end of the presentation, except to acknowledge the efforts of the EPOC Steering Committee and to thank Justin from Ionis, who helped me with some of the slides on terminal sterilization, and to thank you guys for your attention.
Thank you, Daniel.
Thanks, Brian.
Thanks very much, Daniel, for your presentation. So our third presentation now is from Pavel Wadomski, is the Senior Director of Global Reg Affairs at BioNTech. Pavel works in the BioNTech's Global Reg CMC disease team and leads the lifecycle management of Comirnaty vaccines. He brings over 12 years experience with global CMC regulatory for both development and marketed products, numerous biologicals and small molecules and various dosage forms. And his talk today is the industry perspective on mRNA technology. So Pavel, floor is yours.
Thank you, Brian. Thank you for a nice introduction. I hope you can hear me well. I would like to thank the European Medicine Agency for organizing this conference and giving me the opportunity to share the industry perspective on mRNA technology. Next slide, please. In my presentation, I would like to provide an outlook on mRNA class of pharmaceutics and highlight opportunities in applying the platform approach, at least from CMC perspective, as well as share consideration on the potency testing. Please move to the slide 4. mRNA technology offers a broad, powerful, and versatile toolkit to combat diseases. More than a decade of mRNA research has led to effective toolbox for future medicines. In recent years, can you move to slide 4? Yes, please. In recent years, several mRNA formats has been developed to encode various proteins. This has required the development of suitable delivery systems that protect the mRNA from degradation and enable cellular uptake and release into the cytosol. As a result, several mRNA delivery formulations can be used. Next slide, please. mRNA-based products hold great potential to treat several disorders, including but not limited to cancer or infectious diseases. Various therapeutic applications may require variable approaches, including different mixtures of multiple mRNA constructs in a single product or multiple antigen constructs. Next slide, please. As an example, a multi-agent approach, I would like to mention off-the-shelf mRNA immunotherapy, targeting a fixed combination of shared antigens, which is currently being investigated for the treatment of cancer. Another example is individualized mRNA cancer vaccine, encoding up to 20 patient-specific tumor neoantigens, aiming to induce a potent and precise immune response against the patient's unique tumor. The mixtures of multiple RNA constructs may have other applications, not only in oncology, but also in treatment and prevention of infectious diseases. These different approaches present opportunities, but also challenges from CMC perspective. I will discuss some of them on the following slides. Next slide, please. mRNA molecule consists of hundreds of thousands of nucleotides that are linked in a unique order to convey the necessary information to manufacture the corresponding protein in cell. Compared to oligonucleotides, mRNA have a specific structural component such as 5' cap, polyethyl and its untranslated regions, which increase the intracellular stability of mRNA and its translation efficiency in the immune system, leading to lower doses needed to induce a significant immune response. Next slide, please. Before moving to further discussions, I would like to provide some definitions to aid understanding. mRNA, please one more slide. mRNA-based products are those that use exogenous mRNA to express a biological active protein. Within this class, we can distinguish groups or mRNA platforms that use mRNA format in combination with a specific formulation. There may be some aspects that are common to all groups or mRNA platforms, and therefore may apply to the whole class of mRNA-based products. Next slide, please. Prior knowledge is a well-established concept that is clearly referenced in some ICH and EMEA guidelines. The use of prior knowledge can support new product development, process design, and control strategy. BioNTech is building a platform knowledge for different mRNA formats and different formulations used in different clinical programs. Based on our experience, we aim to leverage prior knowledge from products using the same type of mRNA format and formulation. Next slide, please. All mRNA-based products are chemically similar and all are manufactured by the cell-free enzymatic process, commonly called in vitro transcription, using DNA template, RNA polymerase, and nucleotide building blocks. Similarity of the manufacturing process brings potential benefits. As an example, the same set of starting and auxiliary materials can be used for manufacture of different mRNAs. Therefore, prior knowledge can be applied in definition of the quality specification of these materials. Next slide, please. For mRNA-based cancer vaccines, tumor-associated antigens are encoded on mRNA, which can be a single antigen mRNA, monovalent, or multiple antigen mRNA, multivalent. For each new cancer vaccine sequence, only the open reading frame is changed. To date, this platform has been applied to more than 30 antigens. Essentially, the same process is used for all of these RNAs, with the only a few process parameters directly influenced by mRNA length and sequence. This mRNA platform represents mRNA sequence space for the specific mRNA format with corresponding process space. Next slide, please. To enable faster development of new candidates within platform, we propose a concept for product development covering a defined formulation with a corresponding formulation and process space. Based on available CMC data, BioNTech considers the lipoplex and lipid nanoparticles formulation as formulation development platform. What does it mean? For lipoplex platform, data show that this platform can be used for mRNA of different length without impacting the quality parameters. For LNP platform, the manufacturing process is well understood and is considered as robust, resulting in consistency of the quality of different scales. No adoption of process parameters and specification are therefore required when using the same process for different mRNAs. Next slide, please. We would like to propose a concept for process validation covering mRNA sequence space with the corresponding process space. Sequence space can be built based on a dataset collected for various mRNA sequences. In this concept, we assume that only one validation run can be performed using the corresponding sequence-dependent process parameters within the predefined process ranges. After successful validation, all new sequences for the same RNA platform falling within the sequence space can be manufactured without further process validation. Also, in relation to formulation, platform approach can provide supporting data to reduce or waive process characterization and reduce process validation efforts for new mRNA products using the same formulation but different mRNA construct. In this case, PPQ or confirmatory run approach, leveraging previous manufacturing and comparability experience, may be applicable. We also believe that the platform manufacturing process allows for the network validation approach. Next slide, please. Platform approach can be also applicable to the stability of drug substance and drug product. Our experience showed that the dataset of the first sequence within the platform supports the assignment of the initial shelf life for each new sequence, as long as it is within the sequence space. Also for mRNA drug products, shelf life of a new product can be leveraged from the available data once using the same formulation, completed by only confirmatory stability studies. This significantly reduces the development time of a new product. To summarize this part, as shown on the previous slides, internal and external prior knowledge can serve as a basis for building a platform strategy and facilitate faster development and hopefully regulatory acceptance of the next mRNA-based products. Please move to slide 17. However, there are multiple challenges waiting for drug developers. On the next slides, I would like to highlight some of challenges associated with potency testing of mRNA products. We acknowledge regulatory expectations to test potency of biological products. However, in case of mRNA products, there is incomplete clarity on definition and expectation of a potency assay. The potency of mRNA-based products is a complex function of the final product characteristic, including delivery to target cells with an appropriate delivery system and translation of a protein encoding by mRNA. In fact, mRNA products relies on intracellular translation of the antigen in the target cell. However, the protein translated from mRNA undergoes post-translation. Can you move to the next slide, please? However, the protein translated from mRNA undergoes post-translational modification in the cell, and this protein is a bioactive compound. The different steps of mode of action for mRNA vaccines designed to initiate the immune response is presented on the slide. The question we would like to raise is, what is the relevant part of mode of action for mRNA-based product that has to be shown for release and stability testing, as well as for the characterization? Next slide, please. mRNA technology presents a challenge in demonstrating that the antigen is expressed in the cytoplasm of the target cell in structurally integral in immunologically functional form. Structural elements and functional properties of drug substance and the drug product impacting transfection and translation can be characterized and controlled by various methods. Accurate determination of mRNA integrity and confirmation of length and integrity of poly-A tail and untranslated regions of each construct may be used to evaluate the translation efficiency. We believe that the combination of such measurements can serve as a surrogate for in vitro potency assay for multivalent mRNA vaccines, especially when in vivo induction of neutralizing antibodies against each construct is demonstrated in preclinical proof of concept studies. Next slide, please. A complete mode of action panel can be evaluated in characterization studies. Those conducted on physicochemical and in vitro and in vivo level can confirm the structural and functional attributes of mRNA. As a part of the characterization, in some cases, a well-designed in vitro immunoassay can quantitatively assess the potential for a vaccine antigen to elicit the desired immune response. However, a highly selective antibody is needed to enable a sensitive immunoassay. In the absence of specific antibodies against each of the antigens in multivalent vaccines, a combination of biophysical approaches may be needed to mimic potency. Next slide, please. We have already listed a few challenges associated with the potency testing of mRNA products. Amongst others, potential lack of a specific detection antibodies to quantify each translated antigen in potency assay. Difficulty to generate detection antibodies limits the accelerated development potential offered by mRNA technology. Another problem impacting the potency assay could be cross-reactivity of detection antibodies, useful multi-construct products. Another one, especially for more potent vaccines with a potentially lower doses, there is a need for high sensitivity techniques. And finally, setting a clinically meaningful acceptance criterion for a potency assay, especially in case of a patient individualized products, could be a real challenge. Next slide, please. As mRNA technology rapidly evolves, offering multiple opportunities to address the most pressing health challenges and unmet medical needs, there is a need for a dialogue between industry and regulators to address emerging issues. Several of them are listed on the slide. There is a need for a discussion to align on expectation on what is the appropriate part of mode of action to be routinely controlled for more complex mRNA products, how to reasonably address the absence of the specific antibody against of the each antigens in more complex products, if and what combination of biophysical approaches can be considered to determine or monitor structural and functional attributes correlated to potency. What can a matrix of functional attributes can serve as a surrogate for potency testing? And can potency evaluation in characterization studies be a substitute for routine release and stability testing? At the end of my presentation, I would like to highlight that we believe that vaccine platform concept and leveraging prior knowledge significantly accelerates mRNA product development. However, the dialogue and alignment with regulators on pressing issues like potency assay or its potential surrogate may help to facilitate this development even more. I would like to thank all my colleagues who supported me in preparation of this presentation. Thank you for your attention.
Thank you, Pavel. So I think we've had three very detailed and interesting presentations from Rene, from Daniel and Pavel. We're now going to have the panel discussion. So I'd ask if all the panelists could please turn on their cameras. And in addition to the three presenters, I'm happy to also welcome three additional panelists. First, Lubo Nechev from Alnylam Pharmaceuticals, where he's the Senior Vice President of CMC Development. Over the last 19 years, Lubo has led the development and implementation of the CMC strategy for siRNA therapeutics used in 5 approved products. The first of them, Onpattro, was the first approved lipid nanoparticle formulated siRNA, and the second one, Givlaari, is the first approved GalNAc conjugated siRNA. We also have Andreas Kuhn from BioNTech. Andreas has been working with RNA for almost 30 years, starting with his diploma and PhD thesis on the structure and function of small non-coding RNAs. He is currently working for BioNTech as Senior Vice President of RNA Biochemistry and Manufacturing, with a main focus of expanding proprietary technologies to increase the efficacy of mRNA-based therapies and to develop and optimize GMP-compatible manufacturing processes and analytical methods. Then in the room next to me, I have Marcel Hufnagel from the Medicines Evaluation Board in the Netherlands. Marcel's a biologist with a PhD in biochemistry. Marcel is an assessor of biopharmaceuticals, specialized in vaccines, allergens, biosimilars, immunogenicity, and cell and gene therapy products. He's the alternate member for the Netherlands in the Biologics Working Party, and since January of this year, he's the chair of the EMA's Quality Innovation Group, which is a group convened to support the translation of innovative manufacturing approaches to medicines. So we've seen quite some activity in the Slido, and also have some questions that have come to mind during the presentation. So maybe to the new panelists who've joined us first, Lubomir, maybe Daniel's presentation highlighted 3 key areas of starting materials, impurities, and terminal sterilization, which EPOC and others have identified as challenges. Are there any other key areas or topics which you think are a challenge to the industry, and how would you like to see regulators address some of these challenges?
Yeah, I generally agree with the topics that Daniel presented. The numbers there sound very reasonable, although in some oligonucleotides you can achieve lower numbers, but as a general guidance, these sound very reasonable numbers. In terms of starting materials, one that we use, which is not technically a phosphoramidite, is a loaded support, the synthesis loaded support. That's how we introduce the GalNAc. But the general principles that were discussed in the slides are the same, whether it's phosphoramidite or any different starting materials. The fate of the impurities and the purging of impurities, whether at the starting material stage or later at the oligonucleotide stage, is what determines or justifies the use of starting materials. One main topic that was discussed in the field, at least in manufacturing, is the possibility, and I think RNA mentioned this in his slides, is the possibility of having a solution API as classified as API, because currently a lot of solution or pre-formulated APIs are classified as drug product intermediates, which calls for sterile environment and all the complications that a lot of the API manufacturers don't currently have. That will speed up maybe the move of oligonucleotides into prevalent diseases with a large number of patients there. Right.
Thanks, Lubo. Then I suppose let's stick with the synthetic oligos maybe for the first few questions. Maybe I'll turn to René now. In Daniel's presentation, he made reference to the EMA guideline on sterilization, and he raised the point that theoretical risk assessments could be sufficient to demonstrate certain oligonucleotides with, say, a TM less than 110 degrees or aptamers or conjugate to proteins that these cannot be terminally sterilized. Could you comment on this from a regulator's point of view?
I think it's absolutely fair that, for example, it's very clear for aptamers that they are not withstanding these temperature conditions used for terminal sterilization, or also some conjugates to biologicals, of course, and also for as RNA double-stranded oligonucleotides. It's more a question related to single-stranded, for example, antisense molecules, where these are of course generated to be stable, and so some of them also can survive these terminal sterilization conditions, and in these cases sound justification should be provided. I think what's a very important aspect which Daniel mentioned is also the other quality attributes, not only the purity but also some particles and appearance, and so because it needs a sound discussion on the risk benefit and the balance between this. So I think coming back to the question, it's absolutely okay to have a risk-based approach for some classes of oligonucleotides.
Okay, thanks, René. If any of the other panelists want to comment to this, please go ahead. Otherwise, we might move to another question that was raised by Daniel. Again, maybe to René. Daniel suggests a reporting threshold of 0.20% and an ID threshold of 1.0% for most oligonucleotides with doses less than 100 milligrams per day, suggesting this may be appropriate. Could you comment on this from a regulator's point of view?
Yes, I can comment. Of course, this is not any official announcement or something else, but this is more or less regulatory practice and has been accepted for several applications in the past. Maybe the thinking will be changed in the future if other analytical methods. Daniel says no. Okay, no, but at the moment it's current regulatory thinking in Europe. I know that it also has been accepted by other agencies in other regions. So I think it's reasonable. Maybe regarding the reporting threshold, we have also numerous applications also for early programs, early development programs, where a reporting threshold of 0.1 is used. And probably this is reasonable and so, but I think some future discussions are needed. Maybe Daniel, you can comment how does the sum of impurity change if you have a reporting threshold of 0.2 or 1%?
Yeah, we typically actually use we typically use 0.10. You know, that paper was a bit of a obviously a compromise, and many people contributing to that. I don't, you know, I don't have so much difficulty with the reporting threshold, honestly. It just means I have to sum up a few more things and add them to the total. I think the one that's really important is the identification threshold. And really, René, there, I think that really is driven by quality expectations with a big Q. You know, if it really is just about what we should be able to do, when everything works perfectly, then you know, I think 1% is about the best we can hope for. I mean, if you think about a reactive impurity in a starting material, if you had it in every starting material in a 20-mer, and you have 1%, then you have to control that reactive impurity down to what, 5/100ths of 1% in a starting material. So it is quite challenging. And obviously, longer oligonucleotides, it becomes even more difficult.
Mm. But I think together with the overall control strategy, it's more important to avoid this impurity early on. So
Sure.
process characterization of the optimization of the manufacturing process.
Yeah, I mean, definitely. That's the other thing about oligonucleotide impurities in solid phase synthesis. Once the impurity is formed, it's quite difficult oftentimes to remove it. So the best strategy is to avoid it in the first place. But there is only so much you can. You can only analyze starting materials, which are quite complicated, to a certain extent, and you know, controlling reactive impurities down to a few hundredths of 1% is doable. But even if you do that, then really you can only guarantee then, I think, this 1% identification threshold.
Yeah, to that, I mean, of course, ideally you would control the active impurities at starting material. Sometimes, though, to control them to these low levels will take a lot of efforts and increase the price of these starting materials. There are active impurities that you can show that you can remove the corresponding oligo-related impurities after you've used them, and that will relax a little bit the limits that you put on the starting material. So the overall control strategy and fate of these impurities will determine how low you need to go in the starting materials.
Thanks. Thanks for those points. There's a question then in the Slido from the audience. Maybe I'll put this to Daniel then on a related point. So as more and more new building blocks are introduced into synthetic oligonucleotides, so for example, the GalNAc, do you think that an identification threshold of 1% is also applicable to those related impurities?
Yeah, those are a bit more interesting because I think you don't have the multiplicity effect. So I think it's hard to use that argument for sure. Yeah, I mean, perhaps in that case, but you know, then what do you have? You have I mean, the identification threshold is the threshold above, is really the threshold that controls unspecified new impurities, things that are that you haven't seen before. So having more than one, having one for impurities that are derived from GalNAc or some other targeting ligand, and a different one for the oligo, for things that come from the oligo starter materials. I don't know. I'm not sure how that would work practically very easily. So I think you just have an identification threshold, and you know, I really think that sort of 1 milligram number in the Berkey paper is quite important.
I mean, for practical reasons, you probably want to keep the same identification threshold.
Yeah, right.
Oligo impurities that are related.
I mean, the point of the identification threshold is that it speaks to impurities that kind of come as a surprise. I mean, you hope you won't find new impurities.
Yeah.
But again, there's only so much you can control to in an oligonucleotide, and that's why the small molecule limit needs to be adjusted up.
Okay.
I really think it's driven by quality expectations and not necessarily by safety expectations.
Yeah.
So René, would you care to comment to that?
I think it's more useful to have a common identification threshold, and to repeat it, I think the good characterization of these very complex starting materials for GalNAc construction and so on is most important.
Yeah, I mean, René, to your point, by the time you justify the starting material as a starting material, you probably will know the impurities that are related to the active impurities from that starting material in the oligonucleotides.
Okay, thanks. So I think we might.
But Lubo, sorry, there should be, of course, this criticality assessment also for these kind of impurities.
Absolutely, absolutely.
Okay, then I think we might change track a bit towards mRNA. And we have Andres here from BioNTech who's joined us. So maybe a question to Andres. In Pavel's presentation, he talked about monovalent and multivalent products containing mRNA antigens. What specific challenges do you face when formulating multivalent mRNA in a finished product compared to a monovalent formulation?
So I think one of the biggest issues there is identity testing. So I mean, you have to verify that all the RNAs that you want to have in there are really in your product, and that they are in there in the right ratio. So it's a combination of identity and ratio testing. And so that speaks on one side to also manufacturing challenges, of course, establishing a process that makes sure that all your RNAs end up in your formulated product, and then verification of that. I think a second important issue with multivalent mRNA-based products, be it vaccines or be it also therapeutics that are dependent on like multiple proteins, a multimeric enzymatic complex or something like that, is then the potency. And here, I think Pavel alluded to that a little bit, is if you have, for example, I know like prophylactic vaccine is not really the point here, but the example, I think it's quite clear. If you have two very closely related vaccines, like two sequence variants, really to show that both of them are expressed where there's only limited difference in the encoded amino acid and things like that. So I think that are two of the main issues for multivalent mRNA-based products.
Thanks, Andreas. Marcel, do you want to comment on that, or I can give you a next question?
I'll pick it up with your next question.
Okay, so I think the next question I think is one that Pavel posed at the end of his presentation when he described the challenges facing industry to develop potency testing for mRNA products. And the questions he posed were, firstly, can a matrix of functional attributes serve as a surrogacy for potency testing? And if potency evaluation and characterization studies could be a substitute for routine release and stability testing. So I'll ask Marcel maybe to comment on this from a regulator's point of view.
I think scientifically speaking, it's fair enough to appreciate the fact that, let's say, if in characterization we can show the potency and also as Andreas was referring to, if you've got let's say the expression of the one antigen which looks very much as the other one, you could in characterization study them separately and see that they are being expressed, and that would be your characterization. And then having a matrix of, let's say, as Pavel called it, functional assays, which I would call maybe chemical, physical, and immunological assays, would be sufficient, to my mind, probably, if well justified, to serve as a surrogate for potency.
Yeah, if I may chime in quickly, I think what this is really based on is that mRNA is like a way simpler molecule from a functional standpoint than a protein, and that's where like a lot of the potency and biological functionality testing comes from. mRNA functionality, it's dependent on sequence and that you can show at some point that sequence can be translated into the right protein, and then this protein does its function, and that's it. And so the latter part is not really dependent on any characteristic that you can test on the RNAs. You only have to verify that it can be translated, and that's dependent on these parameters like integrity, capping, poly-A-tail, and so on.
Thank you, Andreas. So then let's pick up on a theme that's been running throughout the presentations, and that's the use of prior knowledge and platform technology. And I think, and I've seen quite a number of questions in the Slido around this, and I think it's fair to say we probably have more experience on the use of platform technologies for synthetic oligonucleotides. So maybe René, maybe first if you could give your experience and view on the use of platform technologies for synthetic oligos.
I think it's very reasonable to use some platform approaches, and we have already been contacted in the past by a couple of companies how they can use their platform approaches. There are very, very simple parts of this. For example, some method validation. Should all the methods be product-specific validated, or could a generic validation be applied? And for me, the answer is very simple. And also there are a lot of stability approaches where platform technology can be used because, for example, the antisense molecule, they are designed to be stable, and we have never observed any stability problems also in clinical trials for antisense oligonucleotides. Situation will change, for example, for complex delivery systems and so on, lipid nanoparticles. And if you compare with synthetic peptides, we very often see stability issues with peptides, but not with antisense oligonucleotides. So are some approaches proposed by the companies to use in the future limited stability programs, and with this respect we are relatively open to discuss these approaches, what could be done for stability in the future and so on. These are small parts. for us, it's a question more how to define the boundaries of such a platform, and most important, how to document it. Because it should be a kind of living file, and so should be included in submissions without giving us hundreds of pages. So to find a compromise how to make the change with the authorities is not so easy, but I think we are in a good way, and also we have recently established this quality innovation group at the EMA, and platform technology for oligonucleotides and for mRNA entities is also considered to be a topic in the future.
Thanks, René. Well, then that's a nice segue to Marcel, who's the chair of the Quality Innovation Group. And I think Marcel, in Pavel's presentation, he explained these concepts of an mRNA sequence space with a corresponding process space and how that could be part of one platform. I think it's clear from what we've seen. We've less experience at the moment with mRNA technology compared to the synthetic oligonucleotides. But building on what René has said, where do you see this going with platform approaches for mRNA technology?
Well, as already said, we are open to platform approaches and also using prior knowledge, and all the prior knowledge we could accept has to be justified. But I think for both for mRNA and for synthetic nucleotides, we can accept prior knowledge and use it. Like for stability, it's clear that if you've shown this for the longest and the shortest sequence, then this kind of stability data would be accepted. And also for process validation, we could generally accept this kind of matrixing approach. Of course, you have to take into account to what extent there might be differences between, let's say, two oligonucleotides, having maybe a different nucleotide composition and the nucleotides in the manufacturing process. But generally speaking, I think we would accept a lot of prior knowledge to, let's say, support the characterization of the manufacturing process and also of the product and of the drug substance. Can I add, because one of the things I also saw was about the mode of action between Pavel asked questions about, and of course we've got the nucleotides themselves, the mRNA, and on the other hand you've got the lipid nanoparticles, which to us are excipients. So the drug substance would have, let's say, the mode of action of encoding for the protein, whereas the lipid nanoparticles are there for let's say, getting it to the site where it needs to act. Thank you.
Thanks, Marcel. So then, let's pick up on that point. Then there's a question in the Slido about the LNPs and the lipids, and maybe Lubo, you could address this. The question is, if new lipids are included in LNPs which have not been used in humans before, do they need to be developed like NCEs, new chemical entities? I mean, you have, I think, some experience in Alnylam with this. So could you comment on how the development for such novel excipients is managed?
Well, I can say how we did it. We consider them as novel excipients, and then we characterize it as novel excipients. So yeah, NCE. It's all the excipients GMP manufactured. Just because there is no purification step between the manufacturing of that excipient and then it getting into the patient. Other than that, it's not, I mean, the section is not that much different than a drug substance section in terms of identity and impurity profile and the impurities from the excipients that actually in the lipid nanoparticle. I don't know if that answers the question, but I think the short answer is yes.
Thanks, Lubo. So then we just have a few minutes left. There's a couple of shorter questions maybe I could pose. So, Rene, from the slide, when do you think the draft guideline on development and manufacture of synthetic oligonucleotides will be published for public consultation?
It's hard to predict. At the moment, we have, as I mentioned in the slides, we have two guidelines, one for synthetic peptides, one for synthetic oligonucleotides. We decided to start with this one for the peptides, but for the oligonucleotides, maybe end of the year, beginning of next year. I hope so.
Okay, thanks. Let's work towards that target. I can see in the slide really active contributions, and we've got questions about setting impurity specifications, grouping of impurities, terminal sterilization, prior knowledge in terminal sterilization. So the topics that were highlighted in the presentation are clearly topics of interest, and we've seen a lot. So unfortunately, we won't be able to get to each of those particular questions. I suppose another question I could ask to Daniel, I know there's a number of EPOC working groups. What other topics are you currently discussing, and can we expect further papers to be published by EPOC? And then from that, you know, of the topics you've presented here or are coming, which do you think could have the biggest impact on oligonucleotide development and regulation?
Yeah, I mean, I had hoped to be able to provide the link to the terminal sterilization paper, but it will be in Nucleic Acid Therapeutics. It's in the press. So I think that's a really important paper, and truly it's a really good paper. What else are we working on? I think I wrote them down here. There's a paper on comparability and how to demonstrate comparability. You know, across changes in manufacturing and that kind of stuff. That one is sort of at the beginnings. These are probably things that may come out in the next year and a half, I guess. The comparability one is just sort of getting going. Another one that's just kind of getting going is higher order structure and aggregation. There's one on stability, Rene. One on platform data, which is a little further along, and I think you know, we presented some of that at the DIA last year, Brian. And the last one that's currently underway is microbial control strategy. I think there's those 5.
Okay, thanks, Daniel. Well, there's clearly a lot. Go ahead, Rene.
Just a comment from my side. Of course, these papers are only the current thinking of the industry, but nevertheless, it's extremely helpful to have these paper. Probably, or for some cases, we may have different views, but on a scientific basis, it's really excellent initiative over the last few years, and most of the controversial topics have been published now in such papers, and this is extremely helpful, even if we may have other approaches.
Yeah, I think part of the reason for publishing is just to try to get some dialogue going. And
okay, thanks. Well, thanks, Rene, Daniel. Thanks, Pavel, Andreas, Lubo, and Marcel for very interesting presentations and discussions. I think it's clear in the CMC area there are a lot of topics for discussion, and there's a lot of work going on both at the side of the industry and the side of the regulators, preparing scientific papers and preparing draft guidelines, which we hope to release over the coming year or two for public consultation. So I'd like to thank you all for taking the time for clear presentations and good productive discussion. Unfortunately, we've run out of time and we won't be able to address all of the questions that have been entered in the Slido, but I'd like to thank the participants for being so active and helping to inform the discussion. So I'll close session 2 and I'll hand back to Stefan as our conference chair. Thanks.
Thank you, Brian, and thank you to session 2 panelists and so forth. It was really good discussion here, as you said, looking at the slide, there were many questions for consideration here. So this time it's not just the caffeine level we need to adjust, but also the blood sugar. So there will be a lunch break now for 1 hour, and we'll convene again here 1:30 Central European Time. Enjoy your lunch and see you back. Thank you. Welcome back, colleagues, participants. Hope you had a nice lunch break and is not too much affected by postprandial hypoglycemia and tiredness. So you're ready for the next couple of sessions. I will hand over to Brigitte Anliger from Paul-Ehrlich Institute in Germany to moderate this session. Please, Brigitte.
Good afternoon. Unfortunately, I cannot hear you, but hopefully you can hear me. Okay, perfect. So welcome everybody back to the session 3, the non-clinical session. My name is Brigitte Anliger. I am a non-clinical assessor for gene therapy medicinal products at the Paul-Ehrlich Institute in Germany. And I have the pleasure to moderate the non-clinical session. We will have, again, 3 speakers, 1 from academia, 1 from industry, and 1 from a national medicines agency that will present their views on the opportunities and challenges in non-clinical development of RNA-based medicines. We will have a similar procedure as this morning, so we will have all the 3 talks directly after each other. And after the 3 talks, we will have 30 minutes time for questions and discussions. So the first speaker this afternoon is Hei-Yen Cho. Hei-Yen is an associate professor at University College London, and her research focuses on preclinical development of RNA-targeted novel therapies and translational biomedical research in rare genetic disorders. Hei-Yen is the deputy lead of the novel therapy theme in the NHRA Biomedical Research Center at UCL Great Ormond Street Hospital. And Hei-Yen is leading the RNA therapy program at the Great Ormond Street Institute of Child Health. And she is also the UK representative and management committee member of the European Cooperation Science and Technology on delivery of antisense RNA therapeutics. Thank you, Hei-Yen, for being here today and sharing your academic perspective on the development of RNA-based medicines. And the floor is yours, Hei-Yen. Thank you.
Thank you, Birgit, for the introduction, and good afternoon, everyone. And thanks, everyone. And I'm Hei-Yen Cho from UCL, and thanks, EMA, for this invitation. I will talk about the opportunities and challenges of RNA-based medicine in non-clinical development from the academic perspective. Next one, please. There are many opportunities having taken place to academic in the last decade. The most prominent one is the identification of many new therapeutic targets. The RNA-based medicine has benefited dramatically from the development of advanced genomic technology. For example, here, the different sequencing technology, which have significantly filled our knowledge gap in understanding genetic defects in disease, especially in rare disease, which subsequently provides more therapeutic targets for RNA therapy. One successful example is a 100,000 genome project conducted in the UK led by Genomics England, where many new cases have been diagnosed and lead to further clinical decision-making, including treatment. Just in the beginning of this year, the UK government has announced a newborn screening program with over £100 million funding to Genomics England and the NHS to further boost the genetic studies in rare disease diagnosis and therapy. Next one, please. Equally important opportunity in RNA medicine development is the availability of more new evolutionary platform technology. This includes many new nucleic acid chemistries and modifications, new strategies and mechanisms of action, from siRNA antisense to mRNA and RNA editing technology. and also new delivery system, for example, here, exosome delivery, lipid nanoparticle delivery, peptide, and antibody conjugated delivery for different payloads. This evolutionary next-generation platform technology have provided opportunities to academic and industry, as you have seen from the success of the lead biotechs, as well as many new startups in this area by academics. Next one, please. The RNA medicine has benefited significantly from the development of many new model systems. With the availability of stem cell and gene editing technology, we can create many different model systems now, from somatic cells with easy access, such as in skin, blood, even cells from urine and fallen tooth. They can then be transformed into many other cell types. They can be cultured in 2D for high throughput drug screening, or in 3D organoids for more functional assessment, or even in a more complicated system, such as organ-on-chip for drug development. These human cell originated model systems are particularly important for RNA-based medicine, where many of our products are human gene specific or human mutation specific. So it's very much desirable to have a model system derived directly from or very close to patients. Next one, please. Coming with opportunities, there are also challenges in RNA-based medicine development. The dose-related toxicity is still one of the major challenges that we are encountering in this area, one of our old friends. That means we still need to explore more new chemistry and modification with low or no toxicity. We believe a new delivery method focusing on tissue or cell targeted delivery is another solution to reduce the potential dose-related toxicity of certain nucleic acid chemistry. And next one, please. Efficient delivery of RNA-based medicine to the desired target organs have been a long-standing challenge in this area. As stated in the review from the ECOS-Action Data Consortium, after systemic administration, up to 30% antisense accumulate in the kidneys and around 40 to 50% accumulate in the liver. Tissue or cell targeted delivery of RNA medicine can reduce the non-specific uptake of RNA drug in liver and kidney, and hence reduce the dose-related hepatic and renal toxicity. This technology has been successfully used in several conditions, such as hepatocyte-specific delivery using GANA conjugation, anti-transferrin receptor antibody conjugation for muscle delivery, and the GLP-1 receptor ligand conjugation for pancreatic beta cell delivery. Meanwhile, more cell or tissue-targeted delivery are still needed for many other organs, and is an area many academic labs and industry are working on, including ours. Next one, please. The traditional procedure for new drug development usually starts from the in vitro screening and then in vivo validation and before moving to human trials. But there are several exceptions in RNA-based medicine, as many of this work can also be completed in vitro, such as the backbone platform toxicology, sequence-related off-target effect, part of immunoresponse reaction, and also the efficacy at different levels. Also, many RNA-based medicine, for example, those gene silencing siRNA or antisense or splice switching antisense, their sequence are human-specific. Unless a humanized animal model is available, an unrelated animal model has quite limited application here. So one question often asked by academics is extensive animal study really necessary for some of the conditions, especially for rare disease? Next one, please. I put this slide here. So many of us might have already read this news announced 3 weeks ago that FDA no longer needs to require animal tests before human trials. The law doesn't ban the testing of new drugs on animals outright. Instead, it simply lifts the requirement of using animals to test new drugs before human trials. Companies can still test drugs on animals if they choose to, This news has received quite mixed reactions in social media. As you can see, lots of discussion in Twitter. And while clearly it's a great news for animal welfare organizations, the academic world still keep quite cautious with this change, as usual. Next one, please. The last but not least challenge is the development of personalized or individualized RNA medicine for ultra-rare disease. It is a challenge, but I would also say it's an opportunity for rare disease. Since the report of Milasen, the world's first personalized RNA therapy for Batten disease, which you have already heard this morning from Mika's talk, great effort has been made worldwide by academics, charity, and regulators. In 2019, FDA published the first guidance for the non-clinical testing of individualized antisense drug for life-threatening conditions, which has been followed by many academic groups. Due to the small number of patients, ultra-rare disease are not covered by industry for obvious reasons, and the challenge here is now handed over to academics. So from an academic perspective, we appreciate some clear guidance from regulators on what requirement we should meet in the preclinical studies in order to get approval for this academically clinical trial. It's great to see from this morning's presentation from Dr. Thelma on the draft concept paper from EMA on the development and manufacture of synthetic oligonucleotides. And next one, please. So in summary, this is an exciting time for RNA medicine development, an area full of opportunities and challenges. To maximize its benefit to patients in the future, all parties need to work together, including academics, industry, patient advocates, and regulators. For this reason, I'm extremely grateful of this opportunity and platform provided by EMA to academics for us to give a voice. Next one, please. Next one, please. Yeah. So to finish my talk, I would like to thank many research groups, consortia, and organizations worldwide who have worked tirelessly in this area. Thank you.
Thank you, Haiyan, so much for the very nice presentation and also for raising some important questions, such as the replacement of animal studies and also the difficulties with product development for ultra-rare or personalized medicines, which hopefully will be discussed in more detail in the question and answer part of the session. However, for now, we'll just move on to the second speaker, which is Susan Goody. Susan is a Senior Director and Head of Toxicology at Moderna Therapeutics, where she is accountable for strategies across all non-clinical safety disciplines, including general toxicology, safety pharmacology, genotox, and developmental and reproductive tox. Susan will be sharing her industry perspective, and her main focus today will be the adoption of a platform approach and the leverage of non-clinical platform data. Susan, thank you for being here, and the floor is yours.
Thank you so much for the invitation, and hopefully everybody has not fallen asleep yet after their lunch. While these slides are being transitioned over to my slide deck, I want to thank Haiyan for actually providing a really nice introduction to the non-clinical section. And I think that you'll see some common themes with the industry perspective, particularly with regard to providing, or thinking about what constitutes a data-driven justification for utilizing platform data. Next slide, please. So within the industry, I think we can all agree that there is a deep appreciation for the committed partnership with regulatory agencies such as the EMA to expedite drug development. We all have a common goal of accelerating efficacious medicines safely to patients, particularly those with serious and life-threatening conditions. From an industry perspective, we believe there's opportunities to exist to streamline drug development. And this was a common theme that we heard earlier this morning as well. Samples of such approaches have included proposed platform technology designations that have been outlined in the US in the Prevent Pandemics Act, as well as in the WHO mRNA vaccine guidelines. When we think about platform technology definitions, obviously we're thinking about a technology that can be incorporated into a drug product, such as an RNA sequence, a structure, some type of a mechanism of action or delivery mechanism that's obviously essential for the drug product itself, but something that can be adopted or incorporated and used across more than one drug product. We obviously also have to think about how we can standardize production or manufacturing in order to develop more than one drug. So again, thinking about more than one drug using these same common elements. I think it comes as no surprise to this audience that what we're looking at in terms of benefits are ways that we can accelerate development of innovative medicines, have the ability to provide a faster public health response to crises, particularly think about streamlined regulatory review and resource utilization across different stakeholders, building our confidence in medicines using the same type of platform approach, as well as have innovation incentives. In particular, I'm thinking about cost-effective ways that we can develop next-generation medicines. And as Haiyan so kindly brought up, and you'll see as a common theme throughout the non-clinical section, ways that we can consider to reduce animal use. So let's dive into this a little bit further. Next slide, please. From a non-clinical perspective, some really key definitions that must be agreed upon across the industry is really what constitutes platform components that can be leveraged. We'll get into that in just a moment. The type of platform data that may be relevant or not for each individual drug product, and then the confidence in the scientific rationale for leveraging data. And I think these are three common key concepts that we have to keep in mind. So when we think about our platform components, obviously we're thinking about RNA technology and medicines with three primary components. One is obviously the RNA payload or the cargo. This is, the transient carry of information. Obviously, our bodies process the RNA to elicit some type of downstream effect. So there are going to be some potential common elements of a platform incorporated into these cargo. Delivery vehicle, I think this is probably the one that we can spend the most time thinking about, which is really looking at where the delivery vehicle is oftentimes the primary driver of tissue distribution. It can be a source of toxicological effects on its own, but it is something that is oftentimes one of those core elements that is used across different drug products. And then lastly, we have to think about the downstream biochemical mechanism. So when we think about the RNA payload that's delivered, what's the lasting pharmacodynamic effects of that RNA payload, which may or may not induce toxicological effects. It depends on if it's localization and mechanism of action. So for example, if you have an mRNA that is processed and translated and secreted into systemic circulation, that type of pharmacodynamic effect might be quite different than if you have something that's an intracellular enzyme that is tightly regulated. Next slide, please. Let's think about these proposed definitions. So when we think about the platform components that can be leveraged, we have to think about elements that are integral to the drug composition. And again, we're thinking about things that can be incorporated in more than one drug, and ones that would be without an adverse effect on quality, manufacturing, or safety. So some examples might be delivery vehicles of similar composition, RNA sequence elements that are in untranslated regions, for example. From a drug development perspective, when we're thinking about platform data that can be leveraged, we can think about things such as non-clinical safety data, particularly and especially when the primary effects or the outcomes of a particular study type are expected to be driven by the delivery vehicle. In particular, there are no on-target toxicities predicted by the downstream mechanism of the RNA cargo. Pharmacokinetic data is also another important piece of platform data that could be leveraged, particularly when you're thinking about individual lipid components, characterization of them as a new chemical entity. Biodistribution data also when similar kinetics and tissue residence is expected across different products. So I think when we think about how to leverage these different platform components, once they're defined, what platform components can be leveraged, what data can be leveraged, what helps us build our scientific rationale. First and foremost, the reproducibility and consistency of platform data is something that is incredibly important. We also have to understand the external and internal knowledge about any on-target toxicities that could be predicted by the downstream biochemical mechanism of that RNA cargo. And also, very importantly, we have to think about the clinical plans that are being proposed with these different drug products, because obviously the toxicology data or toxicology package has to be able to support a similar clinical plan. Next slide, please. So from an industry perspective, I think we like to think about decision trees to help us make decisions about what to do. And I think our proposal is to think about a way that we can come to a common agreement about what data can and can't be leveraged. So I've proposed a decision tree here, and let's just start at the top. Where you could work through when it makes sense to leverage platform data from a drug development perspective to help accelerate medicines. First and foremost, the main question that we should start with is, are the on-target effects expected from our RNA cargo based on the downstream biochemical mechanism? Do you expect any on-target toxicities? If the answer is yes, obviously the platform data is not sufficient to leverage. If no on-target effects are expected, the next question should be, are there any common platform elements used across different drug products that could leverage platform data? If there are no common elements, the answer obviously is no, platform data is not sufficient to leverage. However, if the answer is yes, then this would require a scientific rationale, a defendable scientific rationale to utilize that platform data. If the answer to that is yes, then the proposal would be to leverage platform data. Obviously, if the answer is no, it would not be sufficient. So let's think about this pragmatically, about what an example of this could look like. Next slide, please. So if you think about, just as a very basic level, a toxicology package that is being used to support a new RNA medicine. On the left, you could consider a toxicology package when you would do everything for a new drug product. And on the right, it would be when you are able to leverage data. So let's start at the top. When you consider a two-species toxicological evaluation, you first have to think about, okay, in a GLP tox study in rats, if the pharmacodynamic effect of the cargo in rats is not expected, just based on sequence homology, binding, or functional data, and you still have platform rat tox data of a similar design available, one could argue that this provides scientific justification to submit rat tox data from a platform study. Let's move down to the genotoxicity and safety pharmacology studies. In both of these cases, you would argue that provided there's sufficient scientific justification that the cargo is not expected to be genotoxic or induce effects on vital organs, that no new studies would be needed. So you could submit platform genotoxicity studies as well as safety pharm studies to support the evaluation of those components. In this example, in the GLP repeat dose tox study in NHPs, the pharmacodynamic effect of the RNA cargo would be expected, and therefore one could propose that the single study that would be needed to support evaluation of that individual drug product could be the repeat dose GLP study in NHPs. So all told, considering this could be an example of where before leveraging the platform, you might conduct 10 different studies, but after leveraging the platform, you might only have one. Next slide, please. So I think that example can show the potential impact to non-clinical development. Obviously, there's a significant reduction in non-clinical cycle time that can be employed, really expediting novel life-saving medicines to patients. It exemplifies a responsible use of animals, so we're reducing the number or eliminating use in vivo studies altogether. It also allows for reduced test material needed for non-clinical studies. This allows diversion of manufacturing resources to more medicines. And then obviously across stakeholders, we can gain efficiencies in regulatory dossier preparation and referencing, able to cross-reference platform submission documents. Next slide, please. And I might just leave this next slide. This is one that I have a lot of questions. I think for all of us to be considering, thinking about sort of the challenges and opportunities that we have, really thinking about steps that we can partner with the EMA to help identify these platform components, align on such justification for which non-clinical studies make sense to be leveraged, think about strategies that can be used to optimize use of company-specific platform technologies. We're really thinking about that increase in gain of efficiencies for regulatory dossier preparation, and also providing some consistency and clarity on a number of different items. And I think with that, next slide, please. That might be the end of my talk. So thank you very much. Looking forward to the panel discussion.
Thank you so much, Susan, for sharing your views on the platform approach and of leveraging of non-clinical data, which I think definitely needs to be considered by both the developers and the regulators. And also thanks for the discussion topics that you raised in your last slide, which are, I think, definitely topics that we need to discuss and that will also need to be discussed beyond this conference for sure. So in order to have a complete picture, we will now move on to the regulatory perspective, which will be presented by Camilla Svensson from the Swedish Medicinal Products Agency, the MPA. At MPA, Camilla holds the position of a scientific director in pharmacology and toxicology, and with 15 years of experience as a non-clinical assessor at the Unit for Clinical Trials, she gained a lot of specific expertise in advanced therapies and oligonucleotide-based therapies. And maybe it's also worth mentioning that Camilla is a member of the Joint 3Rs Working Party at EMA. So we have today also a regulatory expert for the 3Rs, which stands for the replacement, reduction, and refinement of animal testing. And thank you very much for Camilla for being here, and we are looking forward to your presentation.
Well, thank you, Brigitte, for the introduction, and good afternoon to all of you. Well, Hyann and Susan has indeed put a finger on what the opportunities and key challenges are from a non-clinical point of view, and I think some of you will recognize from what I will say here. But before I respond with the regulator's perspective, I would like to start with the basics on why we need non-clinical data and some notes on available non-clinical guidance. So let's move to the next slide, which is a standard disclaimer, and let's move on to the next one. Thank you. So the purpose of the non-clinical evaluation, well, non-clinical data has an important role in the most early phases of drug development, where information on pharmacological activity, the kinetics, and toxicity provide support for the therapeutic rationale and dose selection, and informs on potential risks and the need for specific risk mitigation measures in the clinical trial. And in addition, at the stage of approval, it provides information for the overall benefit risk assessment and drug label, information that is not obtainable from clinical trials. For example, data on reproductive toxicity potential. But by saying this doesn't mean that all information needs to be derived from new non-clinical studies on the product at hand, but it may be addressed for example, by a combination of using publicly available information and experimental data that can sort of bridge to that, if well justified. So next slide. So what non-clinical guidance do we have available for RNA-based products? Well, there exists guidance in other regions in Japan and FDA, but in the EU, specific guidance for these type of products are currently limited to one reflection paper on genotoxicity for antisense oligonucleotides.
Розумію по-польську, так, але не мовлять надалі.
And it's quite old and outdated from 2005. So we very much rely on the general guidelines we have for non-clinical safety testing, such as the ICH N3 and S6. So indeed, oligonucleotides drugs are mentioned in the scope of ICH S6, which is intended for biologics, but it recognizes the high target specificity and in most cases also high species specificity, and the importance of testing this product in a pharmacologically relevant species, if available. And also for mRNA products, guidance available for gene therapy products should be considered, for example, regarding biodistribution studies. However, RNA-based products, they differ from biologics in several ways. For example, through their physicochemical properties and potential for off-target toxicity. And as Susan also mentioned, complex formulations for delivery. And therefore, most programs largely follows the recommendations in M3, which could mean quite extensive testing, like repeat dose testing in 2 species, reproductive and genotoxicity testing, and rodent cancer studies in some cases. But considering that we also from a regulatory perspective have seen a good consistency in safety profiles between products developed on the same platforms, it seems a valid question to whether it's a full non-clinical program is needed to confirm what's already known for a specific platform. But it's important to recognize that this field is rapidly developing, and with new chemistries and new methods for delivery, this could introduce new risks that needs to be characterized, and some of them are unforeseen. So I should mention here that there is a proposal to introduce a new ICH topic on oligonucleotide-based therapeutics and non-clinical testing. And all that we discuss here today, I hope that will be brought into this guideline if that gets a go-ahead. But I also want to bring into here some ethical and animal welfare considerations. And we do have to take into account 3R perspectives. And the European medicines legislation makes reference to the EU Directive 2010 on the need to replace, reduce, and refine the use of animals in research and also in drug development. So EMA has indeed prioritized these questions now and support the implementation of 3Rs through different activities. And one example to mention here, especially also referring to Hayan's presentation, is the possibility to seek a qualification advice at EMA in order to qualify new alternative methods and introduce them in regulatory testing. Here it's our role as regulators also to ensure that introduction of new methods does not compromise evaluation of efficacy and safety in any way. But the 3Rs should indeed be considered here. So next slide, please. So regarding the opportunities and challenges, I think you will hear that we speak almost in the same voices. I think I can fully agree to what has been said regarding the opportunities. with the knowledge that we do have on some platforms, it should be possible to leverage this knowledge in order to optimize non-clinical program in line with 3Rs. And for products developed then on the same platform. And this could simplify development and assessment of RNA-based products, and that this could go more quickly also to benefit patients with rare diseases. The challenges are high, but not, I think, sort of not overcomable. Can go back one slide. I will stay on the opportunities and challenges for a while. Well, the starting point, I agree with Susan, is to define the platform. And I think you gave some nice examples there. And to include, should the delivery method be included or not, for example. And the more difficult case is perhaps to define what degree of changes constitute a new platform. That is more difficult. I don't have the answer today, but I think we have to work together to that and collect the data and analyze to see, sort of map the commonalities and differences in safety profiles with different chemistries. And it's important that these definitions and requirements are data-driven. So I hope that we can work together on that. And regarding individualized products intended for treatment of ultra-rare diseases that has also been mentioned, we do have experience from my agency that it is feasible to go into clinical trials based on a general but well-described data for a platform, and also to have a good algorithm for how to deciding on the RNA sequence. I'm quite hopeful here that it will be able to define sort of the minimal requirement needed for these type of products. As a regulator, I think a challenge for us is to understand the rapid development in new delivery method and to be able to see what is new and what is very similar to a previously used delivery methods, because different stakeholders or companies use slightly different delivery methods. And to see, and of course, there is a need for a development in this area in order to reach new target tissues and be able to treat new types of diseases. But with the new distribution pattern, there's also possibility for new type of findings. So this usually, I guess this would require a more thorough safety evaluation and would be considered a new platform. Lastly, and just also reflecting on what you said, Susan, on when you gave your example to do one study in the non-human primate, if that is the only relevant species to sort of evaluate the pharmacology. We do have the challenge now that we have a limited supply or a shortage in the non-human primate, so that's also something we have to take into consideration and only use the non-human primates when it's really necessary. But if I have a few more minutes, I just want to make a few comments on where we are today. If we can just go to the next slide. Just briefly touch upon the key points that we expect to be addressed in a non-clinical safety evaluation for RNA-based products. Well, it's well accepted that the risk for sequence-dependent off-target toxicity can be investigated using a stepwise approach, using bioinformatics, complemented with in vitro analysis if needed. But regarding on-target toxicity and sequence independent off-target toxicity. I think where we are now, the general recommendation is to also evaluate this type of toxicity in relevant test species. But there are other things we can do to reduce and refine the use of animals used for these tests. And I think the example you gave, Susan, was very interesting, and I can agree with that it would be possible to use a single species and only one species for the chronic toxicity studies, for example, and to leverage other information if you use the same platform. And on the last slide here, also to make note to put a little bit focus on the safety of the formulation conjugation. So that is also included in the endpoints for the general toxicity testing, because we do know that some of these components that are used in for example, nanoparticles are quite stable and have a very long half-life. So of course we're concerned about the long-term safety, but that's also something that should be addressed in these type of studies. And I think it has been mentioned in previous sessions also that interaction with the regulatory agencies to sort of at an early stage to get advice on programs, I think that is really supported and we have good experience with that. So I think we can move to just say thank you here and go on with the discussion. Thank you very much for your attention.
Thank you so much, Camilla, for sharing your views and also the other two speakers. And I think we have a lot of interesting topics that have been highlighting during the talks, which are the replacement or at least the reduction of animal testing, the use of a platform approach, ultra-rare diseases and personalized medicines. And yes, I think it would also be interesting to see what additional regulatory guidance would be beneficial for the non-clinics. Maybe before we start the discussion, I just had a question to Camilla concerning the decision trees that has been introduced by Susan. And I would like to know your opinion on the decision trees, whether this would be something that in your point of view makes totally sense to decide according to such a tree, and especially to the tree that has been introduced, to decide what data can be leveraged or whether they will be needed additional non-clinical testing, and which one would that be?
Sure. Yeah. And of course, I should clarify that this is my opinion and not the whole community of regulators. But I think that Susan's decision tree, the one you have presented here, is a very good starting point. The only thing I would like to comment is it's usually not that easy to make yes or no decisions. And I think you can have gray scales that you can leverage part of the data that you have from a platform and add perhaps one or more studies to be able to make a complete package to sort of cover the uncertainties. So, but it's a very good starting point, and I think I concur with the major outline of that decision tree. So that was just in brief, yeah.
Yeah, thank you. I don't know whether Susan would like to comment.
Thanks, Camilla. And unfortunately, that kind of put you on the spot a little bit to answer what you thought of my decision tree. I think the way I also see that decision tree is almost to evaluate it on I guess, twofold. One is on a case-by-case basis, depending on the study type that you're proposing to leverage. So let's use the example of cardiovascular safety. If your RNA cargo is expected to have some type of downstream effect on cardiac function, you would obviously want to evaluate cardiac safety with a standalone safety pharmacology study with that drug product. Whereas you might have other components with that same drug product, let's say, for example, with genotoxicity, where you wouldn't expect the RNA cargo to have an effect. So I do see that decision tree as something that could be evaluated on a study-by-study basis, depending on what the requirements are for a given drug product, as well as I guess holistically on a company-wide specific basis. I think we need to think about platform approaches really as a company-specific question, and the justification from a company standpoint. I don't see it being something that would be able to be applied, I think, across different manufacturing processes, for example, that are used across different elements.
Thank you for clarifying that. Yeah, I fully understand that.
Yes, I think this clarification is really helpful. And I think if we deal with platform data and leveraging of platform data, I actually also think that it should be at least initially based on the platform from a given company and not necessarily across different companies, because that would complicate the things quite a lot. And I also think that it will mostly, the situation may be not possible to leverage all the data, but a huge part of the data potentially. I have a following question that goes primarily to you, Susan, but also to Camilla actually. And it's a question basically how extensive the platform data should be, especially if you relate it to, let's say, a full package for a given product that you want to develop. And since you have to consider that your platform data may have to be leveraged to different products, where you may have smaller differences in, for example, the dose level or the dosing regimen. So how extensive do you think the platform data should be?
Would you like me to go first, Camilla, or do you like to answer?
Go ahead.
So I think this is a very unsatisfactory answer, which is I think it depends. I think that it depends on, number one, if it's an in vivo study. Obviously, inherently, there's more variability at times with in vivo data. And I also think that I think if it's an in vivo versus in vitro study, I think those can weigh into the replicates that you would need to do. And so, for example, I think you would also need to have a similar study design. Brigitte, you brought up a very good point about the consistency in the study design to be able to make an apples to apples comparison across different drug products to say that you do have the same type of downstream toxicological or pharmacokinetic effects. So I do think there is merit in knowing that the designs are similar. I think it's hard to put a number on the number, let's say, for example, the number of in vivo studies. Let's use that as an example. Would we need 3? Would we need 6? Would we need 2? I think it depends on what you know about that platform within your individual company, whether it's the same lipids or different, but just what you know in general across the board. So it takes into consideration the totality of the knowledge. Makes our jobs quite difficult because we're trying to piece together data from across different sort of bases. But I do think it depends on what you know, if it's in vivo versus in vitro. But Camilla, I'm very curious to hear your stance on this too.
No, but I'm thinking about the same thing. It's very hard to set the number for how many times you must have tested the new platform. But it will depend on how different this platform is from or similar it is to an established platform. So I think it's also a gray scale there. If it's similar and you see sort of similar type, but maybe a little bit different, that could require less extensive testing than if it's a completely new platform for delivery, for example. So I think you have to take many things into account. But I should say that I wish that data would be more accessible so that you can really make use of the knowledge you have on previous platforms and to sort of leverage that to the new platform, but then add on and you sort of get a map and you can really see the variance, what happens if you make small changes, and you can get safety profile that is within sort of a close to the previous ones.
Thank you.
How I see it. Yeah.
Yes, for the clarification. And maybe I can explain a little bit more what I meant. So it's always a bit of discussion if you have, for example, an mRNA packaged in lipid nanoparticles. Depending on the size of the mRNA, it may be that the size of the lipid nanoparticle is changing as well. And then the question is whether this may affect the biodistribution of the lipid nanoparticle. And so the question is basically how to deal with such a issue. So would you start from the beginning with a platform approach where you then evaluate different sizes of lipid nanoparticles or different sizes of charcoal mRNAs in order to have right from the beginning a kind of a complete picture concerning the biodistribution depending on the size of your particle? Or would you just start with the size that you think you will start your clinical development and then basically risk a little bit the issue that later on there might be some question arising if you have, for example, a smaller mRNA, whether the data can still be leveraged or not? Could you comment on this as well?
I think that's actually a really important point. And the confidence in the data at being able to predict the outcome would be dependent, obviously, on what information is going into the algorithm to predict the data, right? So if you have a range of, let's use your example of lipid nanoparticles that have evaluated in terms of toxicology, and you see a similar consistent response, then you can predict that the outcome would be very similar regardless of the size of the lipid nanoparticle. I think your question about where do you start is a really valuable one, because the question can be, are you really characterizing only the size of the lipid nanoparticle? Is there any potential outcome of the cargo itself on those downstream mechanisms? So you almost have to have something that's you're really trying to characterize from a holistic perspective. As long as you have the range covered, I could see, like in the platform data, I think that could be supportive of a platform approach. Camilla, I'd be curious your thoughts on that as well. That's similar.
But I agree with that, and also that non-clinical sort of evaluations are not isolated, and we of course need quality data to communicate with the quality section to fully understand the critical parameters there, and because that also makes us aware with if there are specific risks that we have not been aware of. But I think in the first case the companies need to address these types, and I think, as I understood in one of your slide, that the structure and other aspects of the cargo is an important aspect in being able to leverage. But if we can map and get understanding, if you make differences in which includes size, of course, of the cargo, how that impacts the safety profile. That could make it's more easy to make use of previous knowledge.
Yes.
But very good point, Brigitte.
Thank you so much. I think we can change now a little bit the focus to the reduction and replacement of animal models. And I have a question from the slide that goes to Hei-En. The question is whether you agree that human in vitro models can reduce or replace animal studies for assessing efficacy of antisense oligonucleotides. But what about the safety assessment of oligonucleotides? Do you also see a possibility there to kind of reduce the animal testing with in vitro models, or what is your opinion on that?
Thanks, Bridget, and also thanks for the question. It's quite an important question. Actually, to many of us, animal studies have been an important and crucial part in our work and has been considered the best way to examine complex conditions and also to determine if our products are safe or effective for market approval. I can see how important it is for farm to perform this. So to consider that question, it's but I do like the idea. So from an academic perspective, what we can do to reduce, instead of just to remove completely, but we can consider how to reduce the number of animal studies. For example, we talk about just now, Susan and Camilla talked about the platform data. That's something quite important. And if a shared platform data on safety profile, that definitely can reduce the number of animal studies. So that's one thing. And also another thing is some other data from using the other advanced in vitro model, for example, from this, iPS differentiate human cell, those data, so you can test the efficacy in those cellular model quite close to what happened in vivo in patients. So those data can also support and help to reduce the number of animal studies. So that's something I read the questions. I was thinking, so what we can do to contribute to this area. Definitely, we support the idea to reduce animal work. And Camilla and Susan, you can chime in.
Maybe I can also add here, because of course these type of models are used in the exploratory setting, but when going in replacing the formal regulatory safety testing in animals, it's of course important that these type of models are validated and shown to be fit for the purpose and robust. And for these there are guidelines and also guidelines available at the European Medicines Agency. And you can seek, as I mentioned in the presentation, a qualification advice to get regulatory acceptance. So I encourage, if you have a method that you wish to get accepted, you should definitely seek regulatory advice on that. But I should also say that we are quite far from having these kind of methods to replace that. So it will be in many years. So what we can do is, of course, to do our best to reduce and refine the testing that we do. Thank you.
Thanks so much to both of you. I have another question that I think goes primarily also to Hei-En, but also the others can, of course, comment. Which is a question from Slido, and it's, are there structural mRNA motifs to facilitate organ-specific expression, for example, in the brain, in the kidney, in the bone marrow? Can you comment on this, whether there are some structural mRNA motifs that you are aware of to kind of facilitate organ-specific expression?
So I need to understand the question a bit better more. Structure mRNA motif.
Yes, I think there will be ways to kind of target the mRNA by the delivery vehicle. I think we are all aware of these possibilities. I understand the question that this goes beyond that, whether there would be some additional ways to target the expression to a specific organ, or actually the translation.
I believe you're referring to regions with an untranslated region of the mRNA sequence, and I would say that the answer is most likely yes. There could be sequence elements that could be incorporated to try to either repress or promote expression in certain tissue types.
I could also share some experience from our group. So, for example, the single-cell sequencing data we use a lot to identify the organ-specific, for example, organ-specific receptor expression in order to develop this cell-specific delivery. If you found the receptor, and then you found the ligand to deliver. So yes, so those information is extremely important, and that there are existing there's a structure mRNA. You can say mRNA or single cell transcriptomic data there to support this study.
Thank you so much. I think we are close to the end of this session. Maybe one quick question to all of you. What is your opinion on the proposed or on the FDA decision that animal models will not necessarily be needed for drug development? So what's your personal opinion on that? Maybe start with Susan and then go on to Haiyan and Camilla.
I think in every case, it would need to be a data-driven decision. I know that seems to be a reoccurring theme in my responses, but I'm encouraged by that point, because there are many cases where you could think about oncology vaccines, for example, where there's not necessarily a pharmacologically active model in terms of an animal species. So I think that's very encouraging because it offers an opportunity to propose an alternative testing approach for demonstrating safety.
Thank you. Haiyan?
Yes, I would like to. Yeah, the microphone is on. So we use animal model to answer questions about in vivo efficacy, biodistribution, and safety. So we will ask the same to the alternative non-animal testing model, and expect they can answer the same questions or even in a more accurate way. So then, yes, we can replace it. However, we have to keep in mind that every model has its limit. A model working for disease A doesn't mean it will work for disease B. This is similar to animal model, that for different products, we will have to use different animal model. Yes, I'm quite positive with this decision, but I think there are lots of work we need to do.
Thank you. And Camilla?
I think we already have opened up in the European region for this situation. And there are, for some very human-specific drugs, the possibility to accept that you do not perform any in vivo studies because they are simply not relevant. So I think it's good that the FDA is on the same page for a harmonized view. But as Susan also mentioned, it should be case by case and a data-driven decision.
Okay, perfect. Thank you so much. With this, we are at the end of the non-clinical session, and I would like to hand it over to Stefan. Thank you.
Thank you very much.
Thank you very much.
Thank you, Brigitte, and thank you to Haiyan, Susan, and Camilla for for this and this very interesting discussion on platforms. I'm pretty sure this is not the last word we will have on platforms in this meeting. In the interest of time, I will pass on the word to Ralf Herold sitting next to me. Please, Ralf.
Thank you very much, Stefan. So welcome to the Scientific and Regulatory Conference on RNA Therapeutics. My name is Ralf Herold from the EMA's Task Force Regulatory Science and Innovation. This is the session on clinical aspects, and these are decisive questions for medicines, from safety to efficacy, to value in healthcare, to value for the individual person. We want to cover primarily clinical research, so the translation into clinical trials, dose finding, iterating between clinic and laboratory, toxicity and long-term safety perhaps, where clinical research is specific to RNA therapeutics. The first three speakers will take us through their views on challenges of the past, of the present, and of the future with RNA therapeutics. And after 30 minutes, I hope we can have some time for a question and answers session. For this, I already invite the audience to think about your questions and insert them into the Slido. And the hashtag is as before, EMA-RNA-QA. I repeat, EMA-RNA-QA, as for the previous session. It's now my great pleasure to introduce you to the speakers. The first speaker is Professor David Henschel, affiliated with the RCSI University of Medicine and Health Data Sciences in Dublin, Ireland. Your research, David, is in the area of neuropharmacology and in particular therapeutic RNA. David, you are working with the International League Against Epilepsy and also have been a coordinator of the EPI-MyRNA Consortium under the 7th framework. Please, over to you. The floor is yours, David.
Okay, thank you. Very good afternoon, everyone. I'm a basic scientist based at the Future Neuro Research Centre at RCSI in Dublin. I've worked with pharma and also biotech over the past 15 years or so to develop antisense-based RNA medicines for neurological disorders. And today I'm going to give you my view as an academic of the challenges and mainly opportunities of moving from preclinical studies to early clinical trials. Next slide, please. So in the next 10 minutes or so, I'm going to introduce the brain disease epilepsy as an example and a rather unique example of how we can begin to test RNA medicines in new ways. I'm also going to introduce a little bit the perspective of patients living with epilepsy on their views on RNA medicines, and then go through opportunities and challenges, and then finish with a short overview of my future perspectives. Next slide. Epilepsy is one of the most common neurological disorders. It affects more than 50 million people worldwide. And the main symptom is recurring seizures. Patients with epilepsy are treated principally with small molecules, anti-seizure medicines, or anti-epileptic drugs. These all work in quite a similar way. They basically dampen down brain excitability, but they don't treat the underlying pathophysiology. So none of these treatments are disease modifying, and we also have about a third of patients for whom anti-seizure medicines just simply do not work. So we need new treatments in the future to target that underlying pathophysiology, and RNA medicines offer us the best opportunity, in my opinion, to move to a new type of therapy for epilepsy. Next. There are two major populations that might benefit from RNA-based medicines. The first are people who have severe drug-resistant epilepsy due to a genetic cause. So we now know that there are both loss and gain of function mutations in a variety of neurotransmitter receptor systems, as well as signaling pathways. These could be targeted directly with antisense oligonucleotides. And we're beginning to see the first of these reaching clinical trials. So Stoke Therapeutics has an antisense oligonucleotide which increases the expression and translation of a sodium channel. And there will be numerous opportunities to use antisense oligonucleotides for patients with these types of mutations. The second major population are those who have acquired epilepsy as a result of a brain injury. This almost never involves a single mutation. There is complex pathophysiology. We see multiple pathways activated within brain tissue from people with this form of epilepsy. But nevertheless, there is evidence that we may be able to use RNA medicines to target even this very complex form of pathophysiology. Next slide. In our research center, and I think this is becoming increasingly common, we're beginning to ask patients what they want, what they look for in new treatments in the future, and perhaps what are their major concerns. Well, we know, and this has come up earlier in the conference, that patients are comfortable with taking RNA medicines. We have Spinraza, for example, for neuromuscular condition. This is, of course, delivered currently intrathecally. We've also heard today, and I've got the same example, that it's now possible to design an antisense oligonucleotide for a single individual targeting their pathogenic mutation. And because of this, there's a very strong interest in the flexibility of RNA medicines to treat many of the worst forms of drug-resistant epilepsy. Next. So we and other groups have assembled patient groups to guide us in our decision-making around RNA medicines. And actually a recent UK-based survey of patient priorities found that gene therapies and RNA medicines were among the highest priorities for them. Patients really do want these types of therapies. They want the potential for a cure. They want disease modification. They want to stop having to take small molecules multiple times per day. And of course, they want fewer side effects. But they also have concerns with RNA therapies and gene therapies. One of the most commonly viewed or voiced is, can they be switched off? How long will they last? And is there a way to stop them working if I no longer need them? And of course, they want to know, are there ways to take these medicines that don't involve painful intrathecal injections? Next slide. The field of epilepsy has a number of unique opportunities which can help us move from late preclinical testing into early human clinical trials. And we've heard already about the benefits, advantages, and disadvantages of using rodents and animals in this regard. There are a number of translational resources that could be better used in the future to help with this large translational gap. Next slide. One of these comes about as a result of epilepsy surgery. So for many people who have treatment-resistant epilepsy, it is possible to remove a piece of brain tissue from those patients, and that will stop the seizures occurring. We now have the technology and solutions in place that we can keep that brain tissue alive and functional. And we've carried out recent studies by incorporating slices of living human brain tissue with antisense oligonucleotides, showing that we can indeed knock down the target within human brain. This is a powerful resource for us in de-risking the steps before clinical trials. And this type of tissue has also been used, and you can see this in the bottom right corner, for transfecting in viral vectors gene therapies. Next. And we can go one stage further. It's possible to design studies where the RNA therapy or gene therapy is introduced directly into tissue which is planned to be removed as part of routine surgery for drug-resistant epilepsy. So in advance of the removal of brain tissue, an RNA therapy can be injected into brain tissue. That tissue is then later removed as part of the scheduled surgery, and it can be studied for effects on safety and toxicity. And we can also slightly modify this design by creating a longer gap between the injection of the RNA therapy and the surgery. This might allow it to be possible to monitor changes in seizures in these patients. And of course, then if the therapy works, that patient would then no longer go through with their surgery. So these are two important translational advantages and opportunities that we can use to de-risk before we go to full clinical trials. Next slide. And next slide. I'm going to mention two other potential opportunities in this area. We've heard a bit about animal studies, and we've mostly been talking about mice and rats. But there's also a species that could be used to de-risk RNA medicines in the future for brain disorders, and that is domestic dogs. Certain breeds of dogs have high rates of intractable epilepsy, and unfortunately many of them have to be euthanized humanely. By working with veterinary colleagues, we may be able to test some of our RNA medicines in the future in this patient population, and we're actually currently engaging with a group in the UK to test an RNA therapy in dogs with drug-resistant epilepsy. Again, this is an opportunity to use an actual disease model rather than a large animal such as pigs or non-human primates, which are large animals but lack epilepsy. So here we have an opportunity for a large animal model with an actual disease that we want to target. And of course, the therapy may be effective for dogs and become a veterinary medicine as well as a human medicine. Next. Last point to make about RNA medicines is, of course, that we can sometimes use RNAs as pharmacokinetic and pharmacodynamic biomarkers. For example, we may find that the RNA that we're interested in alters its level in biofluids such as blood in a way that tracks reasonably well with what is happening in the brain. So it may be possible to remotely measure target engagement, toxicity, or changes in symptoms by monitoring for levels of RNAs in response to the introduction of our RNA medicine. And I think this type of co-development of an RNA biomarker with an RNA-based medicine may be a sensible approach in the future. Next. We've heard already about many of the challenges with RNA therapies, so I'll go through this slide fairly quickly. We know already that antisense oligonucleotides, when injected systemically, do not pass into the brain. That means that currently we're going to have to administer these intrathecally or directly into brain tissue. We also don't yet know what is the optimal chemistry of the backbone of our antisense oligonucleotides and other RNA medicines that is best tolerated within the brain over long periods of time with the least toxicity and the greatest potency. We've heard already about the potential issues with rodent-specific sequences, which can create difficulties in predicting safety, toxicity, and off-target effects. And finally, the last point, we need to remember that when these RNA medicines go into patients, they will be going into patients who are currently taking medication. In the case of epilepsy patients, this would be anti-seizure medicines. And we must be aware that it may be important to test how our RNA medicine interacts with existing therapies. Next slide. So finally, I'm going to give some future perspectives, some directions that we may be able to go into the future. Next. So we've heard frequently from our patient panels that they have concerns with whether or not we can restrict the activity of gene therapies once they get into the brain. There are solutions on the way for this. A team based at UCL recently published that you can insert a sequence that responds to neuronal activity and then switches on a gene therapy. What this means is that that gene therapy can essentially lie dormant within brain cells and will only activate in those cells in which epileptic activity occurs. The gene therapy then switches on, controls seizures for a period of time, and then when those seizures stop, simply switches off again. The second example has come from the field of synthetic RNA. It's possible to engineer sequences into gene therapies which can sense ambient levels of RNA molecules within the cell. So if you have an RNA molecule, which is a good indicator of the onset of pathology, you can use this to drive the on-off of your RNA therapy. Next. And finally, two last points. Coming back to a point I made at the start, that in the case of epilepsy, we have large numbers of patients who have complex pathophysiology. We need to think about what RNA medicines might be best suited to target this. One of those groups of RNAs are microRNAs. These are sometimes referred to as network molecules because individual RNAs can control levels of dozens and perhaps even hundreds of RNAs. So it may be possible to control networks of genes by introducing microRNA therapies that target them. And finally, and this comes back to the issue of delivery, are we going to be able to tag antisense oligos or other RNA therapies in a way that will direct them towards specific regions of the brain, or even within a specific region of the brain to specific cell types? Thank you very much for your attention. That's the end.
Thank you very much, David. Dr. Henschel, I hope you can hear us now.
Yes.
That's great. In the introduction, I explained that this session is on the decisive clinical aspects, but you have extremely nicely exemplified how the translational research and thinking works. And I want to thank you for spanning this wide range of topics. Going to our next speaker, Dr. Michael Wenger. You're affiliated with the company Biotech, where you work as Vice President for Clinical Development after previous position in other pharmaceutical companies, where you contributed to the development already of highly innovative medicines. Michael, you are a board-certified internal medicine, hematology, and oncology medical doctor. I thank you for stepping in at short notice after a speaker from another company could no longer be available. Michael, the floor is yours, please.
Thank you, Ralph. First of all, for the nice introduction, and also to you and the organizers for inviting us for several sessions, actually. Now, BioNTech is perhaps best known because of the development or co-development of the SARS-CoV-2 vaccine. And so I thought it was perhaps appropriate to start this very brief presentation with an application of vaccines, mRNA vaccines in oncology. Next slide, please. Now, when it comes to oncology vaccines, there's basically two very general approaches. One is nicknamed here a fixed antigen approach, and the other one an individualized approach. So with a fixed antigen approach, we leverage several opportunities that we wouldn't have for an individualized approach. So first of all, it's an off-the-shelf medication that can be used for patients that are in high need of treatment right away. It doesn't require any particular production at the time of presentation of the patient. It's an indication-specific mRNA cancer vaccine that could be used for several types of cancers, lung, prostate, and others. Targeting a fixed combination of shared antigens somewhere in the neighborhood of 2 or perhaps just 1 up to 6 or 7 or more. These have to be by nature non-mutated tumor antigens that are shared across patients. And as said, it's applicable for almost all types of tumor antigens, but not for all types of tumors. This is where the individualized approach comes in. This is what most people would refer to when it comes to cancer vaccines, that a vaccine is manufactured for a single patient for a very individualized type of tumor, which in turn means that tissue of that patient is required, tumor tissue, that needs to be shipped to a manufacturing site, needs to be sequenced, and then reveals somewhere in the neighborhood up to 2, 3, or even 5,000 neoantigens. And then powerful algorithms detect the one sample that, or the several neoantigens that may be best to actually be put into a vaccine. This could be up to 20, and these are always unique to that particular patient. This is applicable to a lot of tumor types, but also not to everyone. So for both of these approaches, there's several challenges. And on the next slide, I've listed some of them. Now, the concept of platforms has been discussed during the course of this day a couple of times. So I'll go very briefly over this. This is a very high-level view of the platform concept. So it starts with the assumption that the safety of mRNA-based medicines is, at least in part, driven by their mode of action, and not by the encoded tumor-associated antigen. As been said in the prior session, that this would probably apply only to a single company's platform, not necessarily across platforms with different companies. But within one company, if the mode of action of the vaccine is actually the same, then perhaps there can be some extrapolations made. Now, clinical and preclinical safety data then can be extrapolated perhaps across platform candidates. Can they also be extrapolated across different disease types, given that a lot of the oncology diseases have very specific side effects during the treatment? And can it be even be looked at beyond? So several questions that we don't have an answer for right now, but we think there's definitely something to the platform concept that we want to pursue. Similar to the dose range for one platform compound that may inform the dose range for other candidates, reducing the lag time when we would look into different dose ranges for similar vaccines in the same platform. Now, there's another aspect that when we talk about vaccines, and already alluded to the SARS-CoV-2 vaccine, leveraging the existing post-marketing experience for those RNA vaccines should tell us something. So if we use parts of the technology before, we should probably be able to use some of these learnings also in other therapeutic areas outside of infectious diseases, such as oncology or autoimmune diseases. Now, another interesting part to where we have some answers to, but perhaps not everything, is dose finding. How does dose finding actually work when there is little correlation between the length of the RNA, aka the mass of the RNA, and its translation? So it's not like the longer the RNA is, the more it gets translated, obviously. But even with similar length RNAs, they're not translated to the same degree. And translation doesn't necessarily yield similar immunogenicity. And immunogenicity itself doesn't mean that the tumor is shrinking as it's supposed to be, right? So dose finding is actually pretty tricky in mRNA-based medicines. Next slide. Now, beyond vaccines, mRNA can be used, of course, in oncology for several applications. Monoclonal antibodies can be produced by injecting mRNA in vivo, but also, as in this case, cytokines. So why is this interesting and why is this important? Those of you that are a little older might remember the interleukin-2 infusions that were given to patients with renal cell carcinoma. And the resulting effect was sometimes nicknamed shake and bake when these poor patients experienced severe side effects from the infusion, including fever and tachycardia, hypotension, and capillary leak syndrome. Now, all of this can be to some degree alleviated by a better pharmacokinetic profile, which is actually achieved due to injecting not the IL-2, but injecting the mRNA encoding for the IL-2. You see on the right-hand side the PK profile. So this results in a prolonged half-life, better bioavailability, and lower and less frequent doses. On the next slide, I've shown an example of how a trial looks like. And I obviously don't want to go into too much detail here for this specific trial on an IL-2 variant. But if you just focus on the middle, there's a dose finding going from dose level 1, 2, 3, etc., which is a typical 3+3 dose finding. On the right-hand side, you see the combination therapy expansions with a checkpoint inhibitor and also with standard of care. So the purpose of the slide is to show that the actual resulting trial looks pretty similar to any phase 1b/2 study you would see also for antibodies and for small molecules. So the challenges really lie in the details, specifically as you've seen or as alluded to already with the dose finding. We can easily define an MTD, but we are actually challenged in finding the best efficacious dose. And this is due to the challenges regarding the translation not being linear correlated with the dose, not so simple. On my next and last slide, you see some of the other challenges, some of which have been alluded to during this conference before. One that Tal in the opening session referred to is the lack of consistent understanding of genomic non-integration of RNA. You might be surprised to hear that we still receive many queries about the need for longer safety duration, inconsistent interpretation of the need of genomic DNA, RNA integration, and such requests. Another point is I alluded to the algorithm that's used to determine which epitopes are the best to be included into a vaccine. This has also been alluded to this conference, but I want to just give this example a little bit more clout here. When a tumor is sampled and the neoantigens that are available for mRNA production are detected, they need to be selected because we want only those that have the highest selectivity, a good binding affinity, and the potentially best immunogenicity. So the AI algorithms that we currently have are pretty good in doing this. How do we know? Because we test several of these also in animal models, but they're also improving. So how can you actually fixate a product that is used for individualized treatment if we think we have a better algorithm along the way? Is this a new product? Is this something that needs regulatory oversight per detail? Do we actually need to fix certain parts of an algorithm or not? Now, lastly, perhaps a bit of an outlook where this is going with mRNA immunotherapy in oncology. We strongly believe that neoadjuvant, perioperative, and adjuvant concepts are definitely a future. The immunotherapy has shown efficacy also in last-line patients, but typically they are probably better working in earlier settings. These typically require very large trials and also novel endpoints. Pathological CR has been used now a few times for registration trials, but it's still a bit in its infancy. So we're also thinking about surrogate endpoints that have not been consistently established. An example is ctDNA as a marker of residual disease in the adjuvant setting. How to apply these consistently across novel therapies is going to be a challenge, but we believe it's a good one and some that we want to pursue. Well, thank you for your attention.
And thank you very much, Michael, for this very interesting presentation. You showed applications of RNA that nicely complement the epilepsy applications and cases that David had explained. So thank you for taking us through those thinking and also for sharing your outlook. I understand from the outlook, for example, that early detection such as of cancers is still relevant, and we are not, that reminds us of also RNA applications for diagnostic purposes and early activities for early detection and perhaps even prevention. I was very intrigued that some of the challenges, such as on algorithms, were already mentioned in the other sessions and come back here. So very good. Thank you for this. I now welcome and introduce Professor Joop van Gerven. You are the chairperson of the Central Committee on Research Involving Human Subjects, CCMO, which is in the Netherlands. And in the Netherlands is the Supervisory Authority for Medical Research Ethics Committees and the Regulatory Authority for Clinical Trials. Joop, you're a neurologist and clinical pharmacist and also a professor of neuropharmacology at Leiden University. Please, the floor is yours. Thank you.
Thank you very much, Ralph, and Stefan and all the organizers for inviting me. And I'm certainly very much enjoying this conference, and I think I learn a lot. As Ralph probably already indicated when he kindly introduced me, I'm not a specialist to any extent in this particular field, but I do happen to chair a committee who is responsible for evaluations of trials with this class of compounds. The CCMO is responsible for the evaluation of both the Part 1 and the Part 2 under the CTR for trials with ATMPs, including these RNA type drugs. We also are responsible for evaluating non-therapeutic studies in minors and incapacitated trial subjects. So this is a slightly different system from the rest of Europe, basically, because we share the responsibility for drug and trial evaluations with the Medicines Evaluation Board. We are responsible for the trials. Medicines Evaluation Board is responsible for drug applications for registration. And of course, we closely collaborate together in our scientific advice and in the evaluation of preclinical information. Now, having this rather awkward combination of being an ethics committee and a competent trial authority has given us quite a bit of an experience. Can I have the next slide, please? With trials in this particular area. I remember the first study being in 2006, and I remember that because it was done at my center where I did my clinical pharmacology research at that time. And since then, we have studied 70 or 71 different compounds, or we've evaluated 70 or 71 different compounds, I must say, in the CCMO. And many of them, of course, have never made it to registration, but for all sorts of reasons. And some of them haven't made it to registration because we rejected the outcomes of the application. So we basically rejected the study. We looked at about 130 different studies with these compounds, and well, it's our strong impression that the number is increasing. There's a huge range of diseases and conditions that seem to be amenable to this therapy. And what I, just in a few minutes, would like to share with you is some of the experience we have in evaluating these trials. Now, I've made a slide here that summarizes all this experience. And as you can see, I made a distinction between the antisense oligonucleotides and the siRNA types of approaches. And there are some others, but that number is certainly increasing. Some other approaches, including the coding RNA and the splicing modifiers. But the bulk of experience is in the antisense and in the small interfering RNAs and the microRNAs. And as you can see, our experience is that there seems to be a difference in, let's say, the ease with which these two classes can be developed based on our, of course, limited experience as being a single nation evaluating committee. It seems like the number of rejections from our own committee is certainly larger in the antisense oligonucleotides than in the small interfering classes. There are, of course, more of those antisense oligonucleotides. They were the first to be introduced into this field. I'll come to some of the reasons for the rejections, but it has been our feeling that this is more difficult for us to based on primarily the preclinical evidence than for the small interfering RNAs. It also seems like the development is more uncertain for the pharmaceutical industry and the sponsors. I must say that the majority the vast majority of these compounds are pharmaceutical-sponsored compounds. I could only find one indication where there was an academic group bold enough to try to develop these compounds, which is really much less than in any other therapeutic area that we are responsible for evaluating. The antisense oligonucleotides seem to fail more often in development than the small interfering RNAs. And this may, of course, be related to the indications. The small interfering RNAs, as you can see, have been registered primarily for indications where the target is in the liver. And I think in the 6 compounds that I've listed here that I could find that were registered in Europe and in the United States, 5 of them were GalNAc conjugates. With the antisense, that is much more difficult. There was one compound registered, also a GalNAc targeted compound, volansorten. But there are much more difficult indications being targeted with the antisense oligonucleotides, which is certainly one of the reasons why this seems to be a more challenging type of compound. Can I have the next slide, please? Some common grounds for deliberation and questions, of course, and sometimes for rejection are listed here. It's basically what I just mentioned is that about 6% of the studies with the antisense oligonucleotides were rejected, and almost always because of insufficient supportive evidence in our view. This is not a European view, of course. This is not a view shared by all European authorities, but it's a view that represents the scientific rationale and clinical experience of our committee members. But it is, and we never make a conscious separation between antisense and silent interfering RNAs. But it often seems like there is not enough, particularly pharmacokinetic evidence for penetration of the compound into target tissues, or for pharmacological effects or molecular biological effects that seem in any meaningful way to be related to the pathophysiology of the disease. And that by itself is one of the major rejection grounds. Another reason is that the antisense oligonucleotides, because of their indications, are often developed, or more often developed in children. Now, I've listed the number of studies in minors. 35% of them are with the antisense oligonucleotides are in minors, and 26% for the small interfering RNAs. The major group seems to be between 12 and 17. There doesn't seem to be much of a difference in the numbers exclusively developed for very small children, but particularly in the Netherlands, there is a very strict legal system for non-therapeutic studies in children. So this may lead to a somewhat larger problem with accepting antisense oligonucleotides in children. The major group of compounds that we rejected in this regard were related to Duchenne's disease. So, can I have the next slide? A major problem seems to be to get the antisense oligonucleotides into the muscle tissue. So we have some recommendations based on this experience. It has been very useful to engage in scientific advice, and in the Netherlands, if you plan to do a study in the Netherlands, this is offered by the CCMO in collaboration with the Medicines Evaluation Board. We particularly welcome academic groups. I think there are special rates for academic groups, but like I said, so far actually none of them have shown up for this particular class of compounds. We also welcome researchers from the industry, particularly when they want to explore new approaches. There are, of course, many different ways of using these technologies, and we want to very much be involved in thinking about the early development, particularly for these types of compounds. And we may also share some of the experience that don't generally come up with regulatory advice, because of course we know about the trials that actually failed. It's important to provide a strong pharmacological rationale. That may be something particular for our committee, but I think in general it will be very helpful for other regulatory authorities as well. The pharmacokinetics, tissue penetration are very important. Very difficult in muscle, apparently, not so difficult in liver, also not so difficult in the central nervous system, but surprisingly difficult in the eye for all sorts of reasons. The pharmacodynamics are very important. I would certainly recommend developing a biomarker along with the early development, preclinical development of a drug, and use that consistently in the early development program. That will not only facilitate our approval of drugs, but it will also, from our experience, facilitate the early stages and predictions for clinical effective doses. And one thing particular to the Netherlands and children, but I think an important message for all studies in children is to avoid studies that are limited to pharmacokinetic safety and tolerability, and at least think about offering a therapeutic dose also to children who participate in the earliest stages of development. So I think that's what I wanted to tell you, and thank you very much.
Thank you very much, Joop. And in particular, thank you for doing the exercise and summarizing and analyzing the clinical trials that has been seen by the CCMO and generating these insights. I believe that's a very important work that perhaps we don't have the time to continue right now, but maybe we can find some way to continue with it also in the ACT-EU initiative context that we're having at the EMA to learn fast and well from conducted clinical research and those proposals for research that couldn't progress. I also heard good news in that you said clinical research with RNA therapeutics is increasing, which means that thanks to the previous sessions that we heard about, there's a pipeline that materializes in proposals for clinical trials. So I'm sorry that in this session we have technical issues, and there's a consequence you'll hear just in a moment. And since we are running a bit late because of that, I would just want to ask one question to the speakers in this session and invite all the speakers to come on camera. Thank you. And today, the clinical session was the shortest session in principle, even though the questions are decisive, as we heard. If you think of the actors in the clinical trial enterprise and the way they are working, the way we are working, what do you think is the most important element that needs to be improved for RNA therapeutics to come through? Just think of any aspect from collaboration, funding. What about platform trials, patient engagement, embedding in public-private partnerships, trial logistics, statistical methods? What's your view? And maybe I can just invite you to say what it is and just give a brief reasoning. And why don't we start with David again, and then Michael, and then Joop, if you want. David?
Yeah, thank you. I'm probably the least qualified to answer this question, but I think the idea of having a platform, a backbone for RNA therapies, which is understood to be broadly or universally safe, that can be very flexible, is the big wish going forwards, because there's so many opportunities for treating the kind of the rare and genetic epilepsies, and I think patients will, as they understand the potential of RNA medicines more and more in the future, they're going to demand it, and they're going to expect and need faster turnaround time between the identification of a mutation that can be treated with an RNA medicine and an RNA medicine for their child. So I hope that through the application of, or the coordination between clinical trial authorities and the type of platforms that are developing this, that we can rapidly accelerate this process.
Thank you. I particularly heard the coordination between the actors, and I think that meetings such as this should lead to that. Thank you, David. Michael, what's your element between the actors that you would like to see being improved?
So public-private partnership is certainly something we are very interested in and have actually engaged, for example, with the UK on a large program fairly recently. I think there's always room for improvement. If I had one wish, it would be that we can become more flexible in determining how to assess value of medicines, how to understand these novel types of treatments that are individualized. The regulatory pathways are still a bit lacking behind in that respect. And we are struggling to do trials rapidly because we feel sometimes there is either not enough understanding, or if there's understanding, there's no legislature that can actually fit these novel medicines. So if it's not a request specifically to the agencies. It's more a general wish that to stifle innovation, it doesn't take much, but to propel innovation, you need to actually primarily be flexible and try to understand if there is a way to out of certain dilemmas. So that would be my main wish for the future.
Thank you, Michael. You're embedding the RNA therapeutics into the bigger picture of fostering innovation, and thank you for that. Joop, please.
Yes, well, you asked many different questions or in one or options in one question, but I will only address one. I think I would like to understand why the academic field is lagging behind relative to the other development platforms. As far as my perspective reaches, of course, which may be limited. There seems to be an obstruction in getting academic research to develop compounds, whereas I think this is a particularly promising area for individualized therapy as well. On the other hand, it's also a particularly promising platform for very common diseases. I mean, all the registered compounds that we've seen, or a large part of them, have started their development for monogenetic conditions. And have now been expanded to wider ranges of applications, and this I think leads to questions about costs and making optimal use of the tremendous potential of these approaches. So I think we need a fairly sort of wide discussion about how these types of drugs can be developed optimally, both for individual patients and from a healthcare perspective.
Thank you very much, Joop. And this is in particular then also opening the perspective on RNA therapeutics, of course, being informed by cases, as we heard, from monogenic diseases, genetic diseases, to, as you alluded to, wishes to also see that huge therapeutic effects could be relevant and should be available for patients who have a frequent disease, possibly, but this requires a broader discussion so that we have a multi-stakeholder understanding how to get there. On this note, I would like to thank you all, David, Michael, and Joop, for the discussion. Also from the audience for the question that I've picked up and that we heard from the speakers. We'll close the clinical session. I hand back over to Stefan with thanks for having supported me during the technical issues here. Thank you.
Thank you, Ralph, and sorry to all the audience for the technical issues, which I hope will not reoccur. Thank you, Ralph, for sharing this clinical session. We have been running a little bit over time, but I need a coffee, but I'm pretty sure others need a coffee as well. So, but in the interest of time, I think we should try to start no later than 20 to 4. So 3:40 p.m. we will reconvene the meeting here. So please enjoy your coffee break. Thank you. So welcome back, everybody. Hope you are refreshed with coffee, ready for the last panel discussion, where we would like to put what we heard in the previously 4 very interesting sessions a little bit more into perspective, meeting the scope of the conference, looking forward. How can we, what gaps have been identified in regulatory science? How can we as a regulatory agency help stimulate the development of RNA technologies and so forth, as already alluded to? We have put together a panel, as you see on the slide on the left-hand side, a lot of previously speakers, so I won't introduce them in the interest of time, but we also supplemented the panel here with 3 new faces, Arjen van Hengel from the European Commission's DG Research and Innovation, with Mariette Driesnen, who is from the Dutch Patient Alliance for Rare Diseases, and have also been involved in the Committee for Advanced Therapies, here as the agency, and Jeske Smink, who both have a former academic background and background in many small pharma companies, now with Silence Therapeutics in Germany. I will start out, after introducing my co-chair for this one, to give the floor to the 3 of you to just have a very, very brief intervention and giving some thoughts, having listened to the day. I'm here today to moderate this session together with Marion from the Dutch Evaluation Board. So welcome to you as well, Marion, and thanks for listening in and being here. So turning back to the panel, Arjen, Mariette, and Jeske, please, a quick intervention, each of you.
Okay, thank you. Thank you for the introduction. It has been a pleasure to listen into this event, because as a molecular biologist, it's really exciting to see how these RNA technologies have developed over especially the last 2 decades. And of course, the impressive success of the mRNA vaccines has enormously catalyzed the field of RNA medicine development. So this has, of course, indicated the fact that there are new opportunities for research and innovation to develop these kind of technologies. And it also highlights challenges that we've heard about today in finding ways to optimize the route towards the patient. So coming from the European research part of the Commission, we've been supporting RNA-based medicines via our work programs for several decades already. And of course, that has been focusing on oncology, HIV, on different aspects, and definitely also, of course, the COVID vaccines. An aspect that was put forward earlier also is the element of data sharing. And I think that is something that we see from our side also as something that's being very important, because it's important to learn from each other, especially in a pandemic that is developing quickly, where there's a lack of evidence, a lack of data. But I think this goes wider, and we have to learn from each other also on the elements that do not work. And I think that's a very important element. So what are we doing to support this kind of technology via funding from the European Commission side? Well, most of our funding is not really focused on the development of technology as such. So we have, for instance, the European Research Council, which is providing support bottom-up for novel ideas, and mostly focusing on individual researchers. And of course, elements like what we're discussing about today, like the RNA technologies, can be proposed to be funded under that scheme. Then we have collaborative research on a transnational basis. So that is what we call our Cluster 1 of Horizon Europe, which is focusing on health research. That is focusing on areas where there's a need, where we see need to have an impact. So it is often even agnostic for the disease area. And it is very much open for the research community to come up with novel ideas on how to address these challenges. And of course, there's also there a direction towards challenges in the area of unmet medical needs. This is in line also with what our colleagues in DG Santé are going forward with on the pharmaceutical strategy, for instance. We have also a specific mission on cancer. I mean, cancer was mentioned several times. So also there's a specific focus on that. And exactly under these kind of topics, that are presented in the work programs of the Mission on Cancer and of our health work program, you will see possibilities where mRNA or other RNA technologies would fit to receive funding. So there's also the European Innovation Council, and that's actually providing funding for specific technologies. And they had last year a call open with two cutoff dates that was focusing really on advancing beyond the state of the art for RNA delivery methods, and also on the preclinical validation of novel RNA therapies for complex or rare genetic diseases. So here you see that there are a number of possibilities. The next thing that I would like to comment on is partnerships, because we see a lot of value in that. So we have this year issued a call for a partnership between the Commission and member states on rare diseases, as well as a partnership on personalized medicine. So also there we aim to combine the funding of member states with the funding of the European Commission to make advances in these areas of rare diseases and personalized medicine. And then last but not least, we also have a partnership with the industry. So that is the Innovative Health Initiative, which focuses on pre-competitive areas. So here there are different parts of the industry, like the pharmaceutical industry, but also the imaging industry, the biotech industry, the vaccine industry, that are putting their forces together. We provide additional funding for the academic and SMEs to, together with the industry, focus on research projects, as I said, in the pre-competitive area. And here there's also sometimes a focus on technology development with a look towards the future of what could be important to advance the field where everybody actually would benefit from. So thank you for inviting me.
Thank you, Arjen, and over to Mariette.
Thank you. Well, I represent people with rare diseases. I have been very impressed by the whole program and the wide range of applications of the RNA therapeutics. I would like to focus on what we as patient organizations and patient representatives can do. I think I would like to stress the importance of providing education to patient organizations, and I would like to mention the UPATI training as a very important training, and I hope this will be kept up to date with these innovative therapies as well. So that is one thing. Another topic I would like to stress is the importance of receiving a genetic diagnosis for your rare disease. I think we're still not have equity in the European Union with access to genetic diagnosis by, for instance, whole genome sequencing. So that is a topic of importance as well, to be able to apply these novel RNA therapeutics for the right indication. Of course, for the rare diseases, the pediatric development is very important. And I would like to mention as well for instance, a project like the Connect for Children that we are doing, an IMI project, so together with pharma, that improves clinical trials in children and make the voices of these children heard in clinical trial design. I think that's a way forward as well to do that and make better trials for pediatric better pediatric trials. Let me see. I think it's very important indeed, the data sharing, and to have good governance, best practices. And this is a topic I work on almost on a daily basis these times. So I look forward to make it work. That we have efficient innovation, efficient clinical trials. Well, thank you. That was my thoughts.
Many thanks, Mariette. And then Jeske, please.
Thank you very much, and thank you having us as SME Science Therapeutics being part of this panel discussion. I think it has been a very exciting day and very nice to hear all the different developments, but also the different alignments. We see, I think, a lot of alignment and similar thoughts between industry and regulators as such. At Science Therapeutics, our SME, and a biotech company already existing for 20 years, where we focus on siRNAs that we target into hepatocytes. And we mainly focus on metabolic disease or on hematology, cardiovascular and complement diseases. And for us as an SME, we would be welcoming more regulatory guidance and alignment between the different member states and between regions on RNA-based medicines. And I think this has already been a big topic already today. And as has been shown today by the different presentations, is that individual RNA modalities have unique properties and characteristics for which application of current small molecule and biologic guidelines seem to be inappropriate, and we would be welcoming guidelines appropriate for these type of modalities and the different type of modalities. For example, considering the complexity of impurity profile of oligonucleotides that has been already also shown today by Daniel Capaldi for the EPOC presentation, it's not always possible to separate impurities, and setting specification for individual impurities might not be applicable for all cases, and grouping of impurities might be more appropriate. But we also would like to focus on that guidelines should be flexible to acknowledge the diversity of RNA therapies and focus on how to do things rather than to set absolute limits. And I think it was very nice to see the overview today of René Turmer showing on the work that is currently being done on the new guideline for oligonucleotides, and to see that there's mainly focus on how one can do certain things and how to justify instead of setting set limits. So as an SME, as you can understand, our resources are limited, so we are therefore very much in favor of a science-led approach that supports innovations with a measured approach to risk. And last but not least, also concerning non-clinic studies, which was also a very exciting topic today. I think also, especially in ADME and toxicity testing, there's an obvious opportunity to reduce unnecessary experimentation. There was already quite a big focus on the 3R principle, and I think oligonucleotides, there are quite a big options of also focusing on that area and to reduce animal experimentation. And I think what's very nice to see as well is that I think there's a lot of opportunity for a lot of discussions between the different stakeholders we have. And we can see that there's a lot of discussion going on between the different pharmaceutical companies. I think the initiative like the EPOC is a very good initiative, would allow also for alignment between different companies, but also to have some discussion with the regulatory authorities. So thank you.
So thank you, Jeske, for your for your thoughts. Now we're moving from your introductions to the panel discussion, and you can also put your questions forward on Slido. And today we heard a number of research consortia of academia, and also Mariette was expressing research of consortia together also with public-private partnership, like with IMI. And often including also patient organizations, and which are very active on RNA therapeutics. And I would like to ask a question first to Mariette and then to Annemieke. So how can these consortia get in contact with regulators, and what can regulators do to help those consortia? Mariette?
Yes, thank you. So you mean also the academic consortia?
Yeah, academic consortia together with patient organizations. Annemieke mentioned a few, and also in this panel session, or the panel session, but the session on the clinical aspects, it was also mentioned. So and also from Joop, he from his story, it was seen that sometimes these consortia or academic groups do not go, or maybe do not go often enough for regulatory advice. So what can regulators do to maybe stimulate this clinical development or non-clinical development for these RNA-based medicines?
Right. Yes. I think, well, from a patient organization perspective, what we have done in the Netherlands is at least on the area of the gene therapies, we made like a platform of patient organizations, and we have regular meetings to discuss developments, and we talk to policymakers. And since I'm from the rare diseases, I mean, we focus on ATMPs, and strictly spoken, the RNA therapeutics are not ATMPs, but I take those along in my work. So this is what we do, and we organize webinars to inform patient organizations on the novel developments. So this is one thing. I am as well in the scientific advisory board of the Dutch I can't remember, but for the RNA therapeutics, where Mika Ansmarus started with several academic hospitals in the Netherlands. I think we should work on awareness, have advice from all kinds of stakeholders to contribute and collaborate together. Yes, thank you.
Thank you, Mariette. Annemieke?
Yeah, I was indeed going to say, well, actually, Mariette is part of our advisory board of the Dutch Center for RNA Therapeutics. And I think so when we set up this center, and also when we started the One Mutation, One Medicine, if we're doing these developments for very rare cases, it's crucial that patients are part of the dialogue, and that they're part of these centers to advise and well, tell it, because who best to tell us what is needed. So I think that is definitely something. And so the interaction with the regulators, I think, I mean, the education is multilateral. I think so when you start the dialogues, we are being educated about the regulatory system, and we educate the regulators about all these different specific rare diseases. And we can't expect the regulators to be an expert on all the 7,000, 8,000 different rare diseases. So we need this interaction to educate each other, and then the patients can best say, well, this is what it is day to day to live with the disease. The academics can give well, this is how we think the ASOs would work, and the regulators can give their input and advice and recommendations. So what we did as the DCRT, we wrote to the Innovation Task Force, and we started an interaction and a dialogue with them. I think the challenge, we came really early because, well, we always heard come early, so we did that. The challenge then was that we didn't have a specific product or a specific case. So we just said, well, we want to develop this for very rare patients, and it's not going to be disease specific, or even it's going to be mutation specific. So in theory, we have 10 different patients with 10 different diseases. That was really difficult for the regulators to grasp. So in the end, after some dialogue, we came up with, well, let's give a concrete example. If this is our case, this is how we would approach it. And then that helped a lot with having the ongoing dialogue. So I think the message is to just approach, and even if it doesn't work out, you can have communication and a dialogue to then have a concrete and constructive discussion. Also, from the One Mutation, One Medicine, we went for formal scientific advice. Of course, that is more formalized, so also a lot more work. But also that was really helpful. And I think the challenge is that as academic institutes, we don't have the expertise and the infrastructure to start doing these things, but the innovation task force is really low threshold. So that is definitely something that even if you don't have expertise with the regulatory system, you can do and you can start. And I think starting this dialogue early is important. And we've heard a lot of discussions from the company, but I know that most academics have very little experience with the regulators. And well, learning from our Duchenne dystrophin failure, it's important to come early and to have this dialogue so you can be proactive and anticipate.
Thank you, Annemieke. Joop, you would like to add?
Yes, well, I think there's one thing that might be worth mentioning, and that is further development of the N=1 trial approach. We don't really have much experience with this in regulatory dossiers, and I think particularly if you look for individualized therapy, this is something that requires more thoughts. So it's very relevant to this particular area, also to cellular therapies. There are challenges, and none of these can be solved without active participation from the patients. I mean, you would probably also have to adapt the trial to the individual patient, and certainly take into consideration that these therapies have a very long duration of action, so it may be difficult to do crossover studies. So there are many methodological issues which I think require more thoughts with all the stakeholders and the patient at the center.
Thank you, Joop. And would like to move to another question that was posed also a little bit by Jeske. You mentioned that more EMA guidelines is needed. I was wondering, because EMA guidelines may take some time, if you would also think about, for instance, question and answer documents that you could maybe adapt easier based on the current knowledge that may change over time. I would like to hear your thoughts on that.
Thanks, Marion. I think actually that would actually be a very good idea, because I think already in this early stage it would be very welcome to have certain guidance, because currently we see there's not so much alignment between the different regulatory bodies. There seem to be sometimes questions arising that might not be so appropriate for our type of modalities. And then I think it would be helpful if we already get some more guidance early on. And I think something like a Q&A catalog will be very helpful, because of course it's clear that such a full guideline takes quite a lot of time. So I think from an SME perspective, that's something that we would definitely welcome. Thanks.
Thank you.
And why not pass that question on to Sol and Marcel, because these are the guys who are not either developing the guidelines or will have to maintain the Q&A catalog. Sol, Marcel, please.
Yeah, thank you. Thank you so much for the question. And, you know, there's always this sort of issue is like, when do we develop guidance? Is too early, is too late? Because also, I mean, you don't want to put, you know, requirements in writing that may, you know, stop certain developments. So I think maybe, yeah, finding something in between, something more flexible, as you say, question and answer, maybe it could help. Obviously, I would always say, please come and challenge the regulators. I mean, even if it's for your own specific case, come through scientific advice, through, you know, even if it's too early, through ITF, and make your proposal. I mean, we have heard today, you know, about the use of prior knowledge, platform approach for all the areas, quality, non-clinical, maybe the use of, you know, alternative non-clinical models, as we heard also in the clinical session. So I would say, you know, in the meantime, you can come and you say, okay, provide justification for your approach. Why do you think the knowledge that you already have that has been applied, or even, you know, knowledge from the literature, if it's, you know, something related to safety and the molecular structure is similar and what you would expect. And I think, you know, that triggers discussions among ourselves. And obviously, then we translate, you know, maybe those conclusions to sort of a document. But in the meantime, I mean, I think it's encouraging that we have heard this morning there are a number of documents in preparation, both from the BWP, the QWP, also on the non-clinical side. So again, if something is still not written, come to us and we will provide, our insights into the proposed approach. But thank you. The need is recognized. Thanks.
Yes, thank you. Well, in this respect, it reminds me a bit before the Committee of Advanced Therapy started, guidance was drafted both on gene therapy and cell therapy products, and also monographs were drafted. And of course, the response was from the field, it was too constrained, the requirements in this guidance. And then if you don't draft guidance, then there is the request for guidance. So I think we can never serve you right. But we are happy to draft guidance and to help. And let's say from the Quality Innovation Group, I really invite everyone to come to us with novel developments, novel technologies, share your concerns and see how it does fit or doesn't fit within our current regulation. And we will be drafting Q&As as soon as we can. And that is really the intention, and that's also what the Quality Innovation Group is there for. And as soon as it's sufficiently mature, of course, we will also draft further guidance in collaboration with QWP and BWP. Thank you.
Thank you, Marcel. And I think it's also important what was mentioned before, to come to the regulator, so you can also use that information to provide better guidance documents. Marcel, you would like to add?
Yes, I'd like to add I think there's not been a case in which there was, let's say, a medicinal product not brought to a patient if it were efficacious and safe, because of any issues with quality requirements or anything. I mean, if your product is justified to be safe and efficacious, and you can demonstrate that you can make it in a consistent way, you will always get it registered. If you don't, please send me the examples.
Your email box, you have a great look at. A question came in on Slido. In case of a platform approach, when can we consider products similar? And especially in case of different combinations or a mix of known or approved backbone modifications. And I would like to ask that question to Susan first. Susan?
I think this is actually a really important question. And I think the characterization, or I guess criteria for what would be considered similar, needs to be established. I think not only on a company level basis, but also from a regulatory perspective. So if you think about, let's just use a very basic example of similarities in terms of impurities related to, let's say, the lipids. I think we would have to say that there would be similarities and no more than, like with a new product, if there's no more than what has already been established in a previous qualification for impurities. So I think each company needs to provide data to demonstrate that the products and the platform is considered similar across different drug products. But then also that criteria needs to be agreed upon from a regulatory perspective as well.
Thank you, Susan. And you were mentioning the regulatory perspective. So maybe may I ask Sol to respond to that or comment?
Sure. I think, again, as we said this morning, maybe it would be still too early to consider a full approach, as a platform approach, because again, even though we have, it's a very good experience with mRNA vaccines or with other products based on, non-coding RNAs that have been approved, but we still need to learn more about the use of other molecules through maybe a different mechanism of action or for other indications. So, but again, I think maybe if not the whole approach, you can use parts of what you already know, like to apply to another product. And if again, for instance, from the quality perspective, you have a very similar molecular structure and only a small change, maybe there are parts of that can be applied, or maybe on how we approach, impurity control or specifications, or even potency assays. So I would say maybe, small parts of, or even for non-clinical studies, we can use that, certainly. I mean, the justification together with the data can be provided. And I think we are open to accept, that platform approach, maybe to particular areas, in the dossier. Thanks.
Thank you, Sol. And we have talked now about platforms for more quality or non-clinical, but also in a later session, there was also a question about platforms more for clinical use and clinical testing. For instance, across different diseases, or if those platforms are maybe only for industry, but maybe also for academia. So I wanted to ask the academia representative, Annemieke, to comment on that.
Well, I think I asked that question, so...
It's not your name here.
Oh, well, I asked a similar question at least. And I think, so I think the thing is, of course, that if a company has a specific proprietary modality or tool, then it makes perfect sense that it's a company specific platform. However, if you're talking about oligonucleotides that are delivered intrathecally into the spinal fluid, that are not formulated in any way, and you use a chemistry that's already approved, I would say that's a different case where you may be, well, you can expect a lot of similarities. So I would say that it would not have to be company specific, but that there would be modality specific. And I think what we need to do also is to just set criteria. So what do we call similar and what do we not call similar? Because I think that we've had a lot of these discussions, but so we have the tool, we have the chemical modifications, we have the target tissue, and I think there's probably, we have to set criteria. So what do we call similar, and when do we consider these platform, or even having these platform discussions, rather than having it generally like for all mRNA therapies, because I think that is just way too broad.
But aren't maybe I'm not understanding the concept fully, so I'm playing the fool here. Aren't there two different, ideas about platforms? One thing, if you as a regulator have recognized a certain repetition of things and confirming that doing it be non-clinical CMC or for that matter clinical results in the same, and that then minor changes in either the non-clinical setup or the molecule, then you can concentrate on that minor change and leave the rest out. So that's the platform from the regulatory eye. But I also understand there is a platform issue around having it as a proprietary thing that you can protect with patents, and you can end up in court and finding about this and getting money for, infringing the patent. So I think we need to keep those two discussions a little bit separate, or at least having both discussions within this frame, because I think we as regulators are used to looking at things from a platform perspective and building on the knowledge. I don't know whether anybody else would like to comment on the broadness of and the utility of platforms, also going outside the CMC and non-clinical. I don't know. I'm sorry for putting you on the spot, Tal. You've been up since early this morning. I don't know whether you still are able to give some input here.
Oh, well, I'm happy to try. Thank you. I as I become more involved in my recent past with very early companies, then, every company is trying to develop its own platform from the get-go. And I think every company understands the proprietary nature or the things that they believe are unique to them. I think what would be most helpful is a consistency of approach on what that framework actually encompasses, when is enough data enough, and a harmonization of that approach so that if we need to do NHP studies, then perhaps we can do them anywhere in the world and not just in particular places, just to give you one thorny example. So I think if I look at it from a company creation, the fact that no therapy at the end has been not approved, I accept that. What we don't know is the stuff that never gets there because it is unable to garner funding early on because it is deemed too difficult and complex. So, there's a negative here. We don't know what we don't know in this regard. As it relates to the clinical aspect, let me just make one last point, which is, again, having to do with education and harmonization. Our ability to study these platforms in the clinic is ultimately a function of a public and regulatory acceptability of the putative benefit risk for these approaches. And we spent a lot of time talking about patient understanding, and I'm glad to see Marietta and hear a patient's voice here, but I would remind us that the public's voice is also critical, and patients, especially in rare disease, become very quickly very educated on the things that are very close to their heart. But ultimately, the people who staff IRBs, are not necessarily of the same cloth, and I think the language we use, the description of the anticipated benefit risk and our comfort or lack thereof of any given platform, it becomes really a crucial point in our communication. There are legitimate reasons why different people are going to have different opinions as to the acceptability, but we should at least use the same language and mean the same things when we use the same words as a basis for having that conversation with the broad public. And I think as it relates to the clinical realm, our ability to run clinical trials, our ability to enroll patients with a wide range of unmet medical needs, and our ability to do that effectively, leveraging data that these patients already have access to, or their genotypes as one example. I mean, if you look, just to expand on this for a minute, I think one of the greatest advances that has come out of the COVID era is actually the UK Biobank and the ability in the United Kingdom to have patients, as part of their routine care, choose to enroll in genetic sequencing and then choose to share that down the road if there's a trial that's relevant to them. And, we're all looking at that. That is a huge, huge enabler. And apparently, if you ask people the right way with the right education, it's not the end of the world. And I would encourage, regulators in Europe to take a deep look at that and think carefully about how we communicate in this domain, because there's nothing closer to that, frankly, than RNA medicines and, by extension, gene therapy that actually requires that public understanding as a critical enabler. Over.
Thank you very much, Talon. Maybe I can twist your question a little bit and pass that on to Mariette, because in Europe we have very strict privacy laws and protection of data. Do you as a patient see them as blocking for sharing kind of information like Tal alluded to, genetic data? The European agency is also very focused on using real-world evidence of big data. Could you see this as a hindrance for the development, and in particular development of therapies in this area? We'll start with you, Mariette, and others.
Yes, I think there are certain, in fact, I totally agree with educating the public. I think, and the UK is quite good. The UK government is quite good at doing that. I saw some nice examples, which are examples for other governments, I would say. So, yes, I think we need to, choose carefully on, for instance, genetic data, small groups. But there are ways how we can approach it. And I think, generally speaking, at least for the rare diseases, the unmet need is quite high. So people are willing to advance research and science. But we need good governance and leadership. And I think with some partnership, there are some good examples of partnerships. So that would be a way to go, I think.
Thank you, Mariette, for your contribution. Arjan was telling before a little about different partnerships, and and I was wondering, so where and how?
Ага, добре, давай тепер зайди до мене.
So I was wondering, a question to Arjan. So where and how can pre-competitive research seek support and funding?
Yeah, so the pre-competitive research and also the pre-competitive research, I think here you're referring to the Innovative Health Initiative, because that is really a collaboration between the industry and the European Commission to fund research, and that is in the pre-competitive area. So there's a possibility to seek funding there. There's actually this IHI, the Innovative Health Initiative, on its webpage has a portal where you can support and where you can propose actually research topics to be funded under this structure, under IHI. So where, of course, there's then a benefit or actually a need for public and private research to work together in a pre-competitive area. Now, if we're moving to more general research, under our work program, we can also actively support projects that are in a competitive area. We also can support clinical research there. So there are lots of funding opportunities which you can find via the funding and tenders portal of the European Commission, where all the open calls are. And as I said, there's also the European Research Council, which funds more bottom-up research ideas. So I would like to also comment a little bit on the platforms, because here we see that the development and the integration of platform technology is really something important, because what we want to achieve is impact with what we are funding. And you can achieve, of course, more impact if you use a certain platform technology that is suitable for more applications. So, for instance, in the area of repurposing, we are supporting the development of platform technology, because that is originally very much focused on one compound that you would like to repurpose. And here we want to use platform technology that you can do that in a broader way. The same goes for rare diseases. Of course, there, if you focus on every rare disease individually, it's going to be an enormous task. But if you can use platform technology, also use artificial intelligence in certain aspects, then I think we can move forward. But of course, it's very crucial to do that in collaboration with the regulators to make sure that the technology that you're using also is useful for the path towards registration.
Thank you, Arjan, for this very useful information. And really nice to see also that the platforms are coming back and that you're also really trying to stimulate that with how you organize the funding and to make it more available for different diseases, like you mentioned as well. There was a question posed on the Slido where I would like to go to next. And that was the agency responses to clinical trial applications indicate that the level of understanding of RNA technologies differs across the European Union. So how are the agencies ensuring that there's a common approach, especially if there's little guidance? And I would like to ask that question to Joop, because it's about the clinical trial applications. So Joop?
A common approach.
Oh, yeah.
Well, actually, we have very limited experience with exchanging information across Europe with these compounds. Of course, that's going to be a major learning phase that we will go through as of before yesterday, I guess. Yeah. So I really very much look forward to it. Of course, we have adopted, I confess, a somewhat idiosyncratic approach. That's the experience that I was able to show you. And I'm not immediately assuming that every other regulator will immediately share our views and our considerations in this regard. partly that's because we have this shared responsibility as an ethics committee and as a trial regulator, which may be different from the perspective of a drug registration authority. It's also because we are devoted to these types of compounds, so that is part of our experience and expertise. I would also like to add. But on the other hand, we certainly have blind spots, and I think one of the advantages of being part of the European community is that we can learn from each other, and that we should engage in discussions, not only here but also on specific scientific advices. And in that sense, it may be useful to add that there is a setup for a simultaneous national advice with other regulatory partners that is being organized by EMA, I guess. So we very much look forward to advancing this field by learning from each other.
Thank you, Joop. So we're sort of 5 minutes and then approaching the end of the panel discussion. And Mariette and Arjan and Jeske, you have not been in the previous sessions, but I would like to really ask you, based on also what you heard today, so what is the main point, you mentioned already a few things, but what is really the main point that we have to do going forward for the coming years for those RNA-based medicines, if you have to choose one? Mariette, could you go first?
Well, what I think is important is to maybe develop common core outcome sets as well for certain types of diseases. So, of course, epilepsy is attractive from an outcome point of view since you can easily measure it. But we should, also as a rare disease community, give some thoughts to have common data elements that we can measure to advance the clinical trials in certain areas. That would be a thing I would like to work more on.
Thank you, Mariette. And Arjan?
Yeah, I would almost tend to say that early communication is crucial here, because this is a field that's very much under development. We see, of course, that there's more interest on the academic side, there's more interest on the industry side, and we have questions that need to be solved in terms of the regulatory pathway. So I think the real crucial thing is now communication between the different parties, get the involvement of the patients also in there, to make sure that we find a pathway that is most optimal. So if we're supporting research and development in these early stages, that it has the highest chance to actually reach the patients in the end.
Thank you, Arjan. So really collaborating together and also really sharing this knowledge that we have to really bring it forward. And Jeske?
Yeah, I can only agree with Arjan. I have to say, I think it would be nice to have very early on communication and especially this alignment. I think especially this regulatory alignment that was already part of the previous question that Joop addressed. I think this regulatory alignment is very important for us to move forward. I think in all the discussions we've seen, there seems to be quite a lot of alignment, but it just has to be put in one place. I think that everyone also can agree to that. And that ties in, I think, with having some guidance, which still should allow for flexibility for different modalities we're talking about. Thanks.
Yeah, thank you, Jeske. And really, I think this conference, my personal opinion, it's really so nice to really share this knowledge today with all the different stakeholders. And I would like to, because it's almost approaching 16:30, and so I would like to thank all panelists for the discussion that we had. And I would like to give the word back to Stefan.
Thank you, Marion, and thank you for co-chairing this session, and thank you to the panelists. We are getting close to the final closing remarks. I will pass on to my co-chair, Sol, and if you would like to reflect a little bit on this very long and interesting day, and then I might say a few words to close off at the end. Please, Sol.
Sure. Thanks so much, Stefan, and thanks, everyone. Very short comments. I mean, indeed, it has been a very intense but very interesting day. I mean, we have heard a lot, and I would like to start echoing Emmer's words in the introduction to the meeting this morning. And it's obviously, we have seen today that RNA-based medicines hold great potential. We are moving forward with the lessons learned from COVID-19 vaccines and also from other products that are on the market. The aim of the meeting was to identify regulatory challenges and ways to address these issues. And we have heard a lot during the different sessions on CMC, on the non-clinical session and the clinical session. So indeed, we are taking note of all those comments, and we will appreciate them and also take them on board to provide guidance in the future or any other document that we may prepare, question and answer or others. So obviously, I mean, there are topics that have been sort of been identified in the different areas, again, platform approaches, how to use prior knowledge. And again, we will have to move forward through before we can provide guidance through scientific advice and with your particular cases. Again, even if it's for clinical trials, please come to regulators early and through scientific advice. Even the clinical trials are nationally authorized, but we can provide an opinion on your approach to quality, non-clinical or clinical studies. So again, I think many presentations have indicated that there should be continuous dialogue, and we cannot agree more with that. We need to continue talking with the different stakeholders, industry, academia, patient organizations. So indeed, I mean, we need this continued dialogue to move forward. So please come to us. Challenge us. That in general, regulators, we tend to be a little bit conservative, but I think we love challenges. So it's the only way to move forward. So come to us anytime and we will be happy and, very interested in your project and to discuss new approaches. So again, very exciting times. And thank you so much. It's been a great day. I mean, great presentations. Thanks to the moderators and to all who put together, the program. And it's been really a pleasure participating in the meeting. Thank you. Thanks so much, Stefan. Back to you. Thanks.
Thank you very much, Sol. I couldn't agree more. This has been a very, very interesting day. So, and sorry for the technical glitch earlier today. We don't know what happened there. Fortunately, it didn't kick us completely off track. So I'll close this session. I will first of all thank all of you who have been involved in making this day happen, all the participants, the speakers. I will also thank all of you who joined this meeting either via WebEx. There has been between 350 and 400 people of different kinds listening in all day, and I know that a similar number of participants has been live streaming the event. So this has really been a success for us. I would, before closing completely, thanking a couple of colleagues here for making this happen. I'm pretty sure Tony Humphries has been instrumental in kicking this off. He's shaking his head, I can see, but that's that in modesty. Falk and Ralph for driving this forward, and in particular, Leonor, Malika, and Aline for making this happen. Without your support and drive, this wouldn't have happened at all. We would have been lost at sea. So many, many thanks to all of you. So by this, I will again thank everybody. I hope you had a fruitful day out of this. Definitely, we have had a fruitful day here. I will wish you a continuous onward, nice day, morning, afternoon, evening, depending on where you are on the globe and in the universe. Many thanks for joining us today, and have a nice day. Bye-bye.